Morning, everyone. We're going to go ahead and get started. Somebody give me a thumbs up if you can hear me. Can everybody hear me okay?
Yes.
Okay,
Yep.
great. Thank you very much. Okay, well, welcome. I'm Suzanne Tomlinson with the GCC. We're glad that you're joining us for our first Rigor and Reproducibility workshop in 2021. As always, we're delighted to host some of the best, most engaging speakers that you'll hear that are going to give you a glimpse into the challenges of rigor and reproducibility and the results and repercussions of non-rigorous science, particularly in healthcare. We're only going to be able to just barely touch the surface on some of these issues, but we hope that you'll leave this workshop with a better understanding of these challenges and have some ideas for how to address those in your own research. I'll also note that while this is our introductory workshop, we do have more in-depth workshops on computing in R, publication and reporting, and a new one focused on imaging. So be sure and watch the GCC news. We'll announce those there when those are available. First this morning, we're going to hear from Drs. Lee Ellis and Keith Baggerly, and I'll turn it over to them in just a minute, but I wanted to quickly cover a few items. First of all, I want to, you to note the chat. We want you to enter questions in that chat all day long during the talks. And also you are able to unmute yourself and ask a question if you'd like to do that. After Lee and Keith's presentations, we'll take a break from 11:20 to 12 just to give you a break for lunch, if you need to take care of your kids, if you're at home, and ask that you return to your computers for our experimental design panel that will start promptly at noon. That's the panel where we're going to address some of the questions that you submitted in advance. Some of these will have audience response polls where we will kind of get a feel for what the audience thinks before we discuss the question. We have some great experts in differing areas of experimental design participating as panelists today. But we know that there's also a lot of expertise within our audience. So we really hope that you'll chime in as quickly as you can in the chat. And again, unmute if you'd like to talk in person. And we'll have great discussions with the audience as well. I would ask that if you're not asking a question, you do mute your microphone, just so we don't have too much background noise. I guess it goes without saying that we won't be able to discuss all the questions submitted in advance or in the chat, but you're welcome to reach out to either me or Dawn by email, and we'll put you in touch with someone who can discuss your specific question if you'd like to do that. Now, it may have happened even as I've started talking. If not, within the next couple of minutes, you'll receive an email from Dawn assigning you to a breakout group. These are for the breakout sessions this afternoon. And for those, we ask that when you enter the breakout room, you leave your microphone and your video on. I really want to encourage you to use this time to network, to meet new folks. You never know how you might be able to collaborate down the road. Now, when you're prompted to enter the breakout room, you need to quickly designate a person to be the group leader. And this will be the person that reports out. When we come back out of the break room, the groups will be called on randomly to present to everybody on kind of what they found. And then also we want you to report in an email. You need to take note of all the ideas that come out of your group and email those either to Dawn or myself. You can send separate emails. There's 2 case studies, or you can send them in one. It's up to you. But again, watch for that email from Dawn, and on the email that you send to us with the takeaways, be sure to distinguish those takeaways from the data management session and those from the publication and reporting session. You'll have a timer in the upper right hand corner of the breakout room screen, so you'll be able to see how much time you have left. We're shooting for about 10 minutes. It's not a lot of time, so and once that's over, you'll be returned automatically to the main meeting. In the email from Dawn, we're going to go ahead and send you those 2 case studies that you'll be working on this afternoon. And we ask that you have read through those before the breakout studies and even before the session if you'd like, just to have read through them once so that you have them in your mind and you can quickly begin discussing once you're in the breakout rooms. And so That's really all I have. Just one more reminder to please keep your microphones muted. And with that, I'm going to quickly turn this over to Dr. Lee Ellis from MD Anderson, who is an incredible speaker and involved in so many things in leadership at MD Anderson, in ethics, and really passionate about rigor and reproducibility in research, likely because he's seen firsthand the problems that arise, particularly in healthcare, when research isn't reproducible. You'll hear from him first and followed by Keith Baggerly. And without further ado, Lee.
Well, thank you, Suzanne, and thank you, Dawn, for pulling this all together. I really wish we could meet in person because it's really fun for us to see the looks on your faces when either myself or Keith present a case and you just cannot believe that people have done some of the things that they have done. So having said that, let me share my screen and then go to slideshow. We had a little trouble this morning. Here we go. Okay. So I've been at MD Anderson for 30 some years. I've been lucky that I've been able to play a role in national organizations and international organizations, and I have seen all kinds of stuff in my career. I am vice chair of SWOG, which is one of the NCI-sponsored clinical trial groups, and I'm also a deputy editor of JAMA Oncology. So I get this broad swath, this broad view of looking at research and questions with research integrity. Keith will tell you some wonderful stories of which he was part of breaking the code, if you will. So without further ado, I will move forward and let's talk a little bit. Now, let's see. Here we go. So I'm not the outline for my talk. Let me minimize my screen here. The outline for my talk is that let's talk about, really why we're here today, and why do breaches in research integrity occur, and what can we do to fix this? Normally, if I have enough time, I like to do a little exercise of find the fraud. I throw up some blots or some figures and see if you can figure it out. There will be a couple of those slides in my talk today, but I won't have time to test you on this today. So at the end of my talk, I hope anytime you give a talk, you want your audience to be entertained one way or another. Some of you may feel angry. Some of you, or a lot of you, may feel discouraged. Some can be invigorated, as I'm going to tell you, you don't have to always publish in Cell, Nature, and Science to make an impact. And I suspect there'll be a little bit of everything that I've listed here on this slide. So I've changed my talk a little bit. We've given this talk a number of times, and I try to stay up to date rather than give the same old talk. And every time I give a talk, I go to the Office of Research Integrity website, which is part of our government in the United States, of course, Health and Human Services. I don't know if you can see my pointer, but there it is. And there are some really interesting things that I saw on there now that I had a little bit of time to sort through that. And I know you can't see this, but hopefully we can make this available to you. And this is readily available on the ORI website. You'll notice that there's special attention to research trainees, but there are a lot of other really interesting infographics that I found very well done. I know you don't always think the government does everything well, but I found this very well done. And I think it's worth a quick look, and they make it easy for you to look through this because they realize that you don't need to read another 30-page, position paper. What you need to do is really see things quickly and easily. So the great resource, Office of Research Integrity, ori.gov. Now, this is my favorite morning email, retractionwatch.com. And you may think that this is a little bit of a hoax. A lot of people talk about hoaxes these days, but this is really led by an academic at NYU, Ivan Aransky, and very credible people. Not all of these emails and websites are from credible people, but this one is. So I just want to talk about a story that came out just about 3 or 4 days ago on the number of retracted COVID papers. I know you want to read each and every one of these, but you can't. But there's 120. I'm sorry. I'd like to go back. Yeah, 126 papers that have been retracted so far as of May 23rd. You realize that people want to get things out quickly. They want to gather data that may not be validated. Keith can talk a little bit more about the integrity of databases and so forth. But this is astonishing. And because people are making recommendations for hydroxychloroquine or whatever it may be to treat COVID or prevent COVID, um, be cognizant that not everything that's written out there is real. And this is probably the largest number of retractions in the shortest period of time on a topic that you will ever see, I hope, in your lifetime. Now, I received this the other day, again, trying to stay up to date. And it shows you some of the things that come out to those of us in academia is that somebody will write a book for me and they will put my name on it and I will get credit for it. And of course, this went in the garbage folder after I took a snapshot of this. So this is about, if my pointer works, Lee Ellis's book that they are planning to write for me. So I guess I may have done a little something to deserve receiving this email, but I will let you know I'm not taking them up on this offer. Another great resource for you to look at data integrity and research integrity is PubPeer. Now, this is real. This is a, I can't recall if it's sponsored by the government or not. I don't think it's a government sponsored website, but you're not allowed to post anything without stating who you are. So there are people who will go through papers and they will find a duplication. I'm trying to point this out on the right. Anytime you see the same colored boxes or circles, that means it's the same thing, just moved to a different location. And here you have a panel where you have all kinds of duplications to fit the investigator's hypothesis. And then you may have some comments that are listed here. And PubPeer will actually email you and say that there's a comment about your paper in whatever journal, Oncogene, et cetera. And we would like for you to comment on this. Now, you are not required to do so, but I would think most of us would like to see what type of comments are being made on our papers. And if indeed we did make a mistake, and I will readily admit that I had a postdoc years ago, an outstanding postdoc, who just copied over some actin bands and put it on several different blots instead of just doing the right thing and doing all the acted on each individual blot, and that was caught by somebody on PubPeer. We looked into it. We apologized for it. We looked at all of our data and said it didn't change the hypothesis, but nobody is immune from somebody finding a little something that should not be there. So this is a self-portrait of Elizabeth Bick. So she is a microbiologist. There are several people in the world of microbiology, Arturo Casadevall and Ferric Fang, who I became friends with as we started to take this journey. She really gets beat up a lot because she has this incredible mind where she can not only remember Western blots or bands from figure 1 on a paper when she's looking at figure 6, but she can remember things that papers that she reviewed or looked at years ago. And then she'll go back to that paper and find that there are duplications all over the place. And you can see here just a couple of examples. Again, the same color means that it's the same lifted figure, if you will. And I believe this is another, she is another resource. I follow her on Twitter. And every morning I find all kinds of stuff, which is much more new, many more than you'll see on PubPeer. So she's outstanding at looking at duplications and manipulations of figures. And this is a paper from Elizabeth Bick, like I mentioned, with Ferric Fang and Arturo Casadevall, who's now head of microbiology at Johns Hopkins. Ferric Fang is out west in Seattle. And what they found is that there are lots of image manipulations that occur. And you can see that these, even with these being noted, we really still have a tremendous number of figure image duplications or manipulations. So these are the 4 websites that I would urge you to go to or even subscribe to, especially retractionwatch.com, where you get something every day. You may be notified by PubPeer of something, but I periodically will go on there if I look at a paper I don't really trust, or if I find a big paper published by an author that I really don't trust. I'll go on PubPeer to see if that author has a number of issues that have been raised. And then there are other people who would comment on that as well, not just the person who spotted a problem and the person who's responding to it, but the journal itself may actually make some comments or send you directly the PubPeer comments. Okay, so let's take a little bit of a deep breath and let's take a deeper dive. If I can advance. So this goes back a number of years, but things really haven't changed. In the field of oncology, and I work at MD Anderson Cancer Center, as I mentioned, the success rate from having a drug which looks great in early or in preclinical studies and is moving into the clinic to phase 1 studies and hopefully all the way through phase 3 studies, the success rate of a promising drug that would actually make it to FDA approval is 5% or maybe even less. And this really has not changed over time. You can look at this middle blot from Nature Reviews Drug Discovery, and you can see that what looks promising at the very beginning, there's only a fraction would be an overestimate of really the number that make it in phase 3. So I just lost my screen here. Here we go. Okay. So this is a problem because in phase 1, 2, and 3 clinical trials, we are trying to help our patients. And if the success rate ultimately is anywhere from 3.5 to 5%, and it also depends on how you define success, then perhaps we need to get a better grip on the data that led to the clinical development and to make sure that data is sound. And that's why we're here today. So I go back to my friend Bob Rudinsky. Bob was quite young at this time. He came to MD Anderson about the same time I did in the early 1990s. He and I were both in cancer biology under the great and late, sadly, Josh Fidler, who was chair at the time, and our mentors. And when Bob left and went to Amgen in 2000, he and I stayed good friends, and I mentioned to Bob that, wow, I'm trying to reproduce all this work on IGF-1 receptor targeting and all this kind of stuff, and everything I read shows it's very promising, but I can't reproduce any of this stuff. And he said to me, and I kept this quote from an email, Lee, do you realize that most of what's published in academia cannot be reproduced? So he told me about his direct report at that point, or his supervisor, Glenn Begley, a physician, but he led drug development at cancer drug development at Amgen at the time. And he told me that Glenn had prospectively collected data from studies done at Amgen trying to reproduce some of these studies. And when he couldn't, he would meet with these people to try to figure out, okay, why couldn't we reproduce these studies at Amgen when that was worked at Harvard or worked at MD Anderson or wherever? So I want to go back to studies that came out a long time ago from one of the world's most respected scientists' lab. And basically, this was a period of time where we used to have magazines. I don't know if everybody here has ever seen a magazine, but we had magazines, and on the cover we said, perhaps we found the cure for cancer. It was just incredible. And even though I had seen all of this data, as a physician scientist, it just looked too good to be true. And we did a number of clinical trials, Dana-Farber, Wisconsin, and MD Anderson, and we found nothing with the drug. You would have to manipulate all the data and try to find a little something, maybe a slight increase in apoptosis rates, but we were far away from curing cancer. So we had a number of publications in the high impact journals, Nature, Cell Science, et cetera, that showed that cancer is just melted away with this drug. So why didn't this translate? And I'm going to give a broader view here and say that it's not just a problem in cancer, but it's a problem in other diseases as well. So if you look at the very top of this slide and you look at some neurologic studies, you will see that some of these um, observations, 0% of the studies that are published on a certain topic could not be reproduced. You look at, again, going back to the neurologic literature, only 2 of 12 papers where they were studying spinal cord injuries could be successfully reproduced. Bayer had actually done a study on target validation in cancer and found similar results to what we found. And our study, and I credit Amgen because they were the ones who kept the database, and Glenn and I got together to talk about this. And sorry, I'd like to return back to the prior slide if I could. Let me see if I can do that. I can't. But anyway, we put out a publication in Nature, which I have to say was quite painful in pulling this together. Had to go back and forth a zillion times. I actually withdrew it at one point. And then finally, Nature let us publish our findings, although we could not get granular, because when Glenn would go and meet with people and say, we tried to reproduce these studies and we couldn't, we really couldn't list the individual papers or the institutes where the irreproducible data came from. So we've actually gotten a lot of feedback about that. People aren't happy with that, but that's just, that was just the agreement that Glenn from Amgen made with the investigator when he talked to them. So if you look across the board, there are other people, as I mentioned in the prior slide, who have shown that most, and indeed it is most of studies that show lots of promise in preclinical studies can't be reproduced in other people's laboratories. And ours was the highest at 89% with 53 studies, but you can look across the board here and a number of people have had this trouble. So Nature actually took a hold of this, even though it was Nature where lots of these papers were published that couldn't be reproduced. And they did a survey, and it was quite an interesting survey. And they asked, is there a reproducibility crisis and how severe? And the majority of people actually said, yes, there is a significant reproducibility crisis, and another 38% that there's they believe that there is a crisis there. So there are very few people out there who actually have full faith in the scientific system. And obviously that's a problem, and that's, you know, takes the wind out of your sails as an investigator, but it's important to note the problem so that you can fix the problem. And this is the way that I look at the problems with preclinical data and data reproducibility. And this also occurs sometimes in the clinic, where not everything is reported the way it should be, even though we have a number of safeguards. And I'll put my SWOG hat on and say, from a clinical point of view, you've got to have outstanding statisticians who are always looking over the investigator's shoulders, et cetera. So the spectrum goes from honest reporting, including reporting negative studies, to people who are sloppy, let's say you know, poor stats or cell line misidentification, to selective reporting, only reporting the positive results and just ignoring the negative results, throwing out a mouse where the tumor didn't shrink, to data falsification, where you're massaging the data, to actually fabricating data, not even doing the experiments, but saying that you did. So there are consequences to all of this, in that at the end of all of this is a patient. And our clinical trials are bound to fail if we are not honest in our reporting, and that includes reporting negative data. We waste time and the efforts of investigators and trainees, and I think everyone in the audience here is likely to be a trainee, and you want to get through your PhD or postdoc, and you want to move on with your life. And if you can't reproduce findings, you could waste months or even years of your training trying to reproduce something which can't be reproduced because it wasn't right in the first place. It's a lot of money, which has a big impact on our funders. Our funding rate right now for R01s is about 9%. It would be nice if we can get that funding rate up, and a lot of that depends on reproducible science. And we lose the confidence of our community. And if we talk about being able to do great things and And we can't because our studies are not based on solid science, and people are less likely to donate for cancer research or research in other areas, et cetera. So Len Friedman, who I referenced in another slide here, had done a study to look at the economics of reproducibility. And basically what he showed was that each year there are $28 billion wasted on irreproducible research. $28 billion a year. And this is a few years old now. That's probably gone up a little bit because I don't think the reproducibility of our science has gone up. Now, let me shift a little bit and move to sloppy and selective reporting. And this goes back to some of the things that Keith will talk about: inappropriate statistics or p-hacking, which I won't go into, cell line contamination or drift, and I'll address this a little later on. And also to recognize that journals don't like negative data. We like to, have showy data. We like to have data that's going to lead to citations, which increase the impact factor of a journal. This is a very big game that's out there, and I'm not sure that as a trainee you get this big picture, but that's why we're here today, to try to give you that big picture. And if journals don't like negative data, principal investigators don't like negative data. So there is selective reporting of laboratory studies, and I just mentioned the importance of having positive results. So if you do studies, or an investigator does studies, and the drug works in 2 cell lines and doesn't work in 8, you'll oftentimes only see the results of the 2 cell lines. But there's something missing here, because you may find a biomarker in the 8 that didn't respond. Maybe they had a RAS mutation or a BRAF mutation if you're a colon cancer doc, or whatever it is. So reporting all of the results is critically important, as long as you did the experiments right and you had an unbiased view of the outcome. Everybody needs publications, including faculty, and we'd still have a publish or perish environment, and it's never going to go away. I mean, all of us would be proud to have a Cell, Science, or Nature paper that was real. And even if you don't have those papers, the next tier of papers or whatever it is important. I realize that during the pandemic, a lot of people may not have published much, and that will be excused. But as we get back online and back into the lab, people are going to expect you to, at the end of the year, to do your annual evaluation and show the papers that you published. So there are labs up in the Northeast. I won't mention the city or the institute, but you can probably figure it out, where 2 trainees may be assigned to the same project, and the one who gets the better data is the one who stays on, and the one who doesn't get the better data is terminated and moves to another lab, probably to be put in the same situation. So we tend to only report the positive data and ignore the negative data. And I hope I'm not depressing people too much. The people who have talked at this course over the last few years have heard this a number of times. They probably took an antidepressant this morning, but for those in the audience, it's a little too early to drink, but I'm sorry if I'm depressing you a little bit. But at the end of the day, being honest with everything that you do will pay off. So this is an article, I believe this came out in Nature, and they talk about the pressure to publish or perish, and this all falls back on us in academia, where we need to be able to look at papers that are well done. Editors need to be able to look at negative studies and say, if these studies were well done, they are informative, and we need to embrace the fact that negative data or negative outcomes are still informative. Funding agencies need to do the same. When you go to study section, you're always looking for just the absolute, excuse me for a second, the absolute best data, but that's not always the case. And sometimes finding negative data, as I mentioned, will lead to a biomarker. Sorry, just a little drink this morning. Not drinking beer yet, I promise. And then at the other end of the spectrum is falsification and fabrication. So let's really talk about misconduct. Do investigators actually intentionally falsify or fabricate data? Give me 1 second here. Okay. So these are some letters that I received, some emails that I received after publishing the paper in Nature with my colleague and friend Glenn Begley. And you can see, especially in the middle of the email, that the PIs that this person worked for wanted the postdoc to falsify data, and they refused to do so. So an honest person was fired several times because they wouldn't do what the PI wanted them to do. And they're very good at covering up their data, and they sabotaged his professional life. Some of the comments were so caustic, I can't even show them. As you can see, I've blacked this out. And you can read the paragraph up above about a little bit more of a background in regards to this poor candidate who really had a hard time and had to change labs 7 times. Moving on, and this morning I saw that I could not animate on the share my screen Zoom, so forgive me for the overlapping boxes here. But there was a paper that was published in Nature back in 2014, and this isn't that long ago, which showed that the microenvironment could convert somatic cells into stem cells. And then several people in the scientific community questioned that. A committee at RICN found that the data had been manipulated and was intentionally misleading. This led to the classification that the investigator committed scientific misconduct. And then the senior RICN scientist, the boss at RICN, committed suicide. So the implications of this were massive, but unfortunately, this is kind of fleeting. It's a fleeting headline. I think very few people here probably at this workshop today are even aware of this, and especially in the cancer world, this was not something that hit the headlines because this was a stem cell paper, but I certainly remember this, and I try to pass this on to other people. So it's very sad, but misconduct occurs, and then other people feel the responsibility for it. Now, I'm not going to talk about this because there's nobody in the world who gives a better talk about this issue and research misconduct than Keith Baggerly, and you're going to love his talk that comes up, I think, in about 20 minutes or so. So you can just see this comes from retractionwatch.com, as I mentioned many times earlier. You will see a 60 minutes segment on this that Keith will show you. And this is a story that, as a clinician and looking at an investigator from a top-notch university, Duke University, it's just unfathomable that this was allowed to proceed as it did. And again, Keith will really entertain you with his talk. This came out from the Chinese government, who did an investigation and found that 80% of the country's clinical trials were fabricated, not just falsified, but fabricated. So you realize that this is not just a problem at Duke or a problem in Japan. This is a worldwide problem, and all of us need to be aware of this as we read the literature. So what's the impact of retracted clinical papers on our patients? And there are very few papers on this. This came out in 2011, even before the reproducibility crisis, if you will, made the headlines. Retracted papers based on this analysis impacted an average of 2,600 patients per paper. So I showed you that the drug approval percentage is about 5%, going from promising preclinical work to the phase 3 trial, or maybe even sooner than that. So when papers were retracted for fraud, 1,500 patients were impacted. So papers can even be retracted, but they could still negatively impact our patients. And this is, of course, of great ethical concern. So there are a number of famous fraudulent papers, especially in the cancer world, but I'll quote a couple others. The breast cancer and bone marrow transplants were presented at the 1999 ASCO annual meeting. This was completely made up. Autism and vaccines, and this really has an impact on some of the vaccine hesitancy we have today, not necessarily for autism, but just because people have this general view that vaccines are bad and bad things can happen. This came from Andrew Wakefield. I'll show you on the next slide some of the things that he did, which were unfathomable. And stem cells and tracheal transplants led to the death of several patients, even though this came from a great institution, one of the world's best journals. Macarena, who was a surgeon, claimed to be able to recreate tracheal tubes from stem cells, and patients did just fine. And this was likewise all falsified data and led to the deaths of several patients. So I'm not going to go through all of this. You can see this yourself and read through this yourself. But as Wakefield was saying that vaccines can cause autism, he was working on patenting his own version of a measles vaccine, which is interesting. This was all about money. And it's absolutely incredible. And as I said, you know, the downstream effects of connecting vaccines and bad outcomes probably has a big impact on us trying to get to the herd immunity that we'd like to get to today. So completely falsified, completely for selfish purposes, but people have done this. And I know people in the audience are probably just can't catch their breath after they see something like this. Okay, so I'm not going to just blame it on those people out there. No institute is immune, and here I list several different issues that occurred at MD Anderson. I've also listed several examples of Harvard having some issues with retractions and irreproducible data, falsified data, fabricated data, whatever. I think I have an animation here, and the point that I'd like to make is that it is our responsibility as principal investigators to keep track of the data in real time. When I was an interim chair of cancer biology, I remember having a candidate come to me and they were not able to meet with their PI until they had basically a cell paper ready to go. That is not how we should do science. We should be meeting in person, by Zoom, by however, every week with our trainees to make sure we keep track of the data and provide input. And it's not about a cell paper, it's about seeking the truth. So we did a survey. I'll give credit to Len Welling, who came up with this idea of doing a survey. I included several trainees on this because this was all about trainees. So we did a survey at MD Anderson looking at the issue of data reproducibility and translating from the lab to the clinic. And what was amazing to me is at a time of where you think everybody's apathetic and everybody's just trying to get through the day, once the survey went out after it was approved by the IRB, we had 240 responses in 6 hours. Now at MD Anderson, that has to be a record for people to be able to stop in the middle of the day and do another survey, but they were passionate about this. And again, credit to Len Welling and taking this through the IRB and doing everything the right way. The results of the survey are listed in the next couple slides. Have you ever tried to reproduce a finding from a published paper and not been able to do so? And as you can see, it's about 50%. So frankly, with senior faculty, they've been around longer. I'll admit I'm a senior faculty member. It was up to 70%. The longer you're around and the more you try to reproduce something, the more likely you are sometime in your career not to be able to do that. But even trainees found that 54% of the time they couldn't reproduce a finding that they found in a published paper. You know, again, peer reviewed, probably several rounds of peer review, repeat experiments, etc. So that's a pretty high number. Were you ever pressured to publish findings of which you had doubt? And that's about 22, 25%. That's a lot when you're pressured to publish findings that you're not very comfortable with, because I go back to the publish or perish paradigm that I've spoken about earlier. Have you noted pressure from a mentor to prove that his or her hypothesis was correct, even though the data you generated may not support the hypothesis? And that's about a third. And are you aware of mentors who require a high impact publication before a trainee can leave the lab? And this is shocking. 50% of people said that they would not be able to leave their lab unless they had a CNS paper, Cell Nature Science, or something similar, probably in the same family of journals. And I found this shocking. And that was just before I published my own cancer cell paper and my first one there. So I certainly didn't adhere to that principle, but lots of people have been a victim of that principle. And again, I'm not going to read through this whole slide. I'll just highlight one point of, what makes people actually, who are honest people, but they're working in a lab where they're pressured. What makes them, give in to the PI's insistence that they have a big time publication. And you can read the orange in the middle once you've gotten through the rest of this. The international trainees also have a visa issue. This was something that I wasn't aware of until I saw this, is that in order for some investigators to stay in this country, they have to be productive. So the PI starts to push them with visa issues. They feel a lot of stress and eventually they do whatever the PI wants. So let that soak in for just a little bit. And you see all the pressure that are on lab investigators, especially people coming from other countries, and especially in this era where it's actually very difficult because of COVID, because of political issues, all kinds of things like that. So lots of reasons why people are forced to kind of massage the data, and I'm being gentle when I say that. So we followed this up with a survey on data reproducibility. Delphine, who was managing my lab at the time, and others in my laboratory helped put this together. And of course, we had the outstanding support of Keith Baggerly to make sure that everything was clean and the stats were done right. So let me move forward again. And most of these were from postdocs. I guess students were way too busy to fill out this survey. And this was, most of the people were in cancer biology, but they came from all kinds of different backgrounds. So this was done within the Texas Medical Center. And sorry, to see if I can go back one. So some of these questions that we asked: how often do you authenticate your cell lines? And then you have about 40% which said never. Okay, I'm going to tell you all that's a problem. How often do you test your cell lines for possible mycoplasma contamination? About 30% said never. Again, a problem. Do you or your lab collaborators perform blinded studies? A pretty high percentage here, approaching 40%. When my team goes back to sacrifice mice, I have 3 people go back there. The PI or the investigator knows from which group the mice came. The other 2 have no idea when they're measuring tumors, et cetera. So I do blinded studies when we sacrifice mice to look at tumor burden. I hope you do something similar. Do you consult with statisticians regarding your studies? No, because you have software and you know how to do it on your own. I can see Keith's expression on this right now. Statisticians can teach you a lot. In the longer I'm around, the more I learn from statisticians and how important they are, especially in the clinical trial development and working with SWOG and the Statistical Center for SWOG out in Seattle, where I have wonderful colleagues, and learn to work with statisticians. Bottom line, you need them. Do you feel that it's necessary to have a first authored paper in a CNS journal when seeking an academic position? And this is over 50% feel that they have to have this high impact publication. Not all your data may be strong enough or impactful enough to get you there, but somehow, if you want to get an academic job, somehow you've got to get a paper in CNS. Do you feel the pressure to publish or perish influences the way you report your data? And over 60% said yes. And I've given this talk a lot of times, and I'm still shocked to see some of these results. Have you ever fabricated or falsified data? Of course, nobody's going to say yes to this, but they've certainly seen it in over 20%, and that's kind of bothersome. If you're going to fabricate data, don't let anybody see you. And have you ever witnessed someone falsifying or fabricating data to complete a project or a paper? Now, you realize that when you get reviews back from a review of a paper, they're asking you for all kinds of crazy stuff. And think about that when you're reviewing papers yourself. And sometimes there are investigators, not in my lab, I don't think, but investigators who will provide the data that are necessary in order to get the paper over the hump and get it published. And that seems to be about 25% here. So why does this occur? And I can make this short. I mean, I've already talked about publish or perish. That's the same for both trainees and faculty. Talked about the importance of high impact publications to get a job. And you can't leave the lab unless you've got a high impact publication. Same thing with faculty. We need to build our bio sketches. Some institutes require you to bring in the vast majority of your salary off of grants, even though you may be teaching, you may be mentoring, you may be doing other things. And it's really nice to walk down the hallway and see your cell paper up on the wall outside the faculty lounge at MD Anderson. So the stature and gratification and promotions occur for faculty when you have a big time paper. So there's lots of causes of massaging the data. And I'm certainly not going to go through this. I would just say that Nature survey that I've referred to before has provided data to support this. And let's talk about high impact publications and the term impact factor mania and what this does to our culture. So as I was interim chair of cancer biology walking down the hallway, I ran into a postdoc one day and we just started chatting. And she was kind of a little upset and she turned to me and said, my mentor just told me, quote, you are nothing unless you publish in CNS, Cell Nature Science, of course. So think about the pressure on that postdoc to provide the data that would get a Cell Nature or Science paper. Going back to my friends, Arturo and Ferric, they wrote a nice letter in MBio, Microbiology Journal, talking about impact factor mania, that these impact factor mania where we are looking to publish in the high impact factor papers with impact factors above 20 or whatever it is, and we'll do anything to support that. And I'll just read the quote, associating the value of research with the journal where it was published rather than the actual value of the work itself. And mania is causing profound distortions in the way science is done that are deleterious to the overall scientific enterprise. And I completely agree with this because people will massage the data to get it up there into the big journal rather than tell the truth, which is, of course, necessary and something I've talked about over the last 40 minutes. So they've developed a retraction index. And what this shows is that the higher the impact factor of the journal, the more likely they are to have a retraction. And of course, that's multifactorial because, you know, more people scrutinizing papers from the New England Journal of Medicine than some of the other journals that are listed here. But it does have, you know, it's a nice graphic, a nice correlation that the higher the impact factor of the journal, the more likely it is that they're going to have a retraction over a period of time. And then I like to use a quote from Confucius, a man who has committed a mistake and doesn't correct it is committing another mistake. This was kind of interesting. A Nobel Prize winner declared a boycott of top science journals. And basically that may be really nice for the Nobel Prize winner, but for the people in his lab who actually may have data that are ready to go to Cell Nature or Science, it's kind of disappointing for them. So there are people who feel strongly about publishing good work in good journals, but I'll go back to the publish or perish principle is that you know, it's nice to have a Nature Cell Science or Cell or Science paper. There's nothing wrong with that as long as the data is real. So my final comment on impact factor mania, it's perfectly fine to publish in Nature. You should strive for Nature, but don't ever lie or die for Nature and never, ever compromise your ethics. So what can we do to fix this as we finish up the talk? So back in 2015, after we published our paper in Nature, I had the honor of giving the John Wayne Clinical Lectureship at the Society of Surgical Oncology. And with that, I was asked to write a review from the Lancet or Lancet Oncology. And I include several of the things that I think we need to do in order to improve our ecosystem, which includes publishing negative findings, and as reviewers, allowing people to publish negative findings, etc. And this should be available to you online. The Office of Research Integrity lists every year the number of people who have found guilty of misconduct. I find it incredible that there's so few people who are actually found to be guilty of misconduct. I just updated the 2021, and I personally know more people that have committed misconduct than are listed here for the entire country. And the question is, are we doing enough to punish these people who have violated our trust? So what are the consequences of being found guilty of misconduct? Well, the paper's retracted. Okay, big deal. You have your research supervised for 3 years, which is something I don't even understand how your research can be supervised. You can't serve on committees for 2 to 3 years. So therefore, you are rewarded for committing research misconduct, but you can still receive NIH funding. So to get to the end of my lecture, if you're guilty of fraud, the punishment must fit the crime. And there's really no deterrent for such bad behavior. And I think the whole system needs an overhaul. If you're found guilty of misconduct, I think that's the end of your career. And I know that's tough, and I'm a surgeon, all that kind of stuff, but no one should ever commit misconduct, research misconduct. And one of the problems is that if someone is accused of research misconduct by going to the research integrity officer at their institute or whatever it is, and there is an investigation, the Office of Research Integrity in Washington does not do the investigation. The investigation is done through reputable and most of the time senior individuals at the institute, but it's done by your own institute. So you can imagine that there is a little bit of a conflict of interest in your own institute investigating your own faculty or postdoctoral fellow or whoever for research misconduct, when if that news gets out, that's bad for the institute. Now, I don't really know a way around this. I'm not sure the government's going to be flying in and going over a bunch of data, but I do note an inherent conflict of interest. And the same thing, eventually the decisions are made regarding what happens to that faculty member at the level of the top officers or leaders at the institute, provost, president, whatever it may be. And again, there's a conflict of interest here because you are trying to protect your institute's reputation. So I don't have time to go into too much of this, but I think it would be nice for the ROI to be more involved in some of these investigations. So if you trust no one at your institute, so for instance, the University of Texas has a fraud and abuse line that you can place your concerns online, and it can be done anonymously, etc. And I'm going to end the talk by referring to a hero of mine, John Lennon. I'll get to that in a second. My friend Mike Fish, who used to be at MD Anderson, is now in Chicago, but he's still part of SWOG. After our paper came out, he said, "You've uncovered a thorny problem in academia, selfishness." And that's quite an interesting way to put this. In moments of weakness or at the extremes, this creates an undertow away from integrity in science and public health. This is the single biggest limitation in the field. And there's two very smart people I'm quoting at the bottom. John Lennon, just give me some truth. I'm looking at this artwork right now in my living room. I'm sick and tired of reading lines by seasick, narrow-minded, short-sighted hypercritics. All I want is the truth. Just give me the truth. And this guy Einstein's pretty smart too. He said, "The right to search for truth also implies a duty. One must not conceal any part of what has been recognized to be true." And I think he's saying you need to report your negative data as well. And this is on the outside of the National Institutes of Science in Washington. And with that, I will end my talk. And if I went over or under by 2 minutes, I can't recall when I was supposed to end. My apologies, but it's a little bit of a crazy time, and I'm glad I was able to get through my slides. Thank you all.
Thank you, Lee. As always, great talk. And we have about 5 minutes for questions. And I've had a really good time watching the chat as though you couldn't see the faces of the people in the audience, I could imagine the faces as people express their frustration in the chat. So we had a conversation going on among some of the attendees, and you addressed some of these, after the questions were asked, but along the lines of Doris was talking about, in her field, she knew a few labs where they had data that was bogus for years. They kept having their papers accepted in Nature, and the people who criticized them had their careers trashed. It went from the bench through clinical trials. She says she lost a lot of colleagues and friends for criticizing the data. And then there's questions she also asked, are there career consequences for retractions? And you certainly addressed that. It's not super satisfactory, what you talked about. Kara has a similar question, and I'm kind of going to say several of these are along the same lines, and you kind of address them all. But how can PIs demonstrate progress to funding agencies to show that they're not wasting their money if it's not through their publications? And so just kind of some real frustration here. Are the scientific journals responsible for promoting and accepting papers that are retracted later on? So you want to just kind of address that, and then I can go on. Like I said, we've got about 4 minutes.
Yeah, I think, there's a lot of different moving parts there. I would say that it starts at your institute and the culture at your institute. And if people got as much credit for being a mentor, an advisor, and at times a psychiatrist to our trainees, especially during this time, if they got as much credit for that as a Nature paper, we'd live in a much better culture. Fortunately, I will credit Peter Pisters at MD Anderson's my old friend. He's my president. Yeah, I know he's not on this call here, but you have a nice person who has been in the trenches, done all these things, recognizes these things, and it all goes up to the culture that you set at the top of your institute. I really can't speak for the other institutes because I don't have a lot of interaction with them. But I think everything starts at the top, and I can guarantee you in my laboratory, I look at every blot every week, even during the pandemic, once our people got back into in lab, and whether the blots were pretty or not, I don't wait for a paper to be ready to be published to look at the data. I look at it in progress. And if my hypothesis is wrong, it is wrong. And that's good, because if I'm right all the time, there's something wrong with, with our culture. So I think these are good questions. As I mentioned, I think the ORI should play a heavier hand because of the conflict of interest within the Institute. But I can say that when you have somebody like Bill Plunkett leading the investigations at MD Anderson, I know he does a thorough job of investigating this, but I'm not sure that I can say that for every institute. And it's not always somebody who is as polished as a Bill Plunkett is at MD Anderson. So there are lots of moving parts here. The bottom line is you are responsible for your data. And if you have negative data, you need to talk your PI into putting that in. It can be in a supplement. I understand, as somebody who's an editor for JAMA Oncology or deputy editor, you don't want to take up precious space in a journal with negative data, but you need to have that in the paper somewhere. And that's why these days with online supplements, you can provide a venue for the negative data as well. However, if the negative data supersedes the positive data, as I mentioned, 2 cell lines it worked, 8 cell lines it didn't, it's really important that you show that in the real paper and not just force somebody to go online for that.
Thanks, Lee. I'm going to invite you to continue answering some of these specific questions in the chat. But before we move on to Keith's, which is about that time, so there's Alex and S. Corvino, expressing some frustration. Everybody knows about this. Nothing changes. Alex saying, why can't publish or perish paradigm go away? And you've kind of addressed it's starting at the top at your institute. But it'd be great to hear you comment, even if it's in the chat, about, this really comes even from NIH, right? Because our papers are our currency. Do you want to just quickly address that and then jump into the chat before we go to Keith?
Yeah, it's really hard to figure that one out. I think it would be really important for the NIH to take on the initiative of saying, okay, we've given this guy 9 guy or woman, whatever, 9 R01 grants. They publish in all the big journals. What has happened to it? And I think this deserves, another
Yeah,
crack at meeting with the folks from the NIH and saying, you need to do a autopsy, if you will, or a survey, if you will, of somebody's 20-year career and what has happened there. And then do you continue, funding them if they've actually done nothing that has been reproduced?
yeah,
This is off the top of my head. I don't know if it's making any sense at this point, but we need to be a lot tighter. We need to pay attention to PubPeer. We need to comment on PubPeer when we see things that aren't right, because the journals look at PubPeer, I can tell you that. And, it's like I tried to tell you, the reputable websites versus the non-reputable websites. If anybody needs to get in touch with me, I've got some other meetings to go to today, but if you want to email me directly for an issue or a question you're having, I'm happy to do so. I'm probably not going to be able to get to all the questions in the chat today. I have another job that I need to do, but I thank you for your interest and attention, and I look forward to Keith's talk.
I think you pretty much addressed all of them. They were all along the same lines, but I want to encourage the audience to be thinking about those questions when we send out the after workshop survey. We really want to hear your thoughts on on ideas of how to improve this at an institutional level and at the NIH level. What would your suggestions be? So thank you, Lee. We really appreciate it. And we'll jump now.
Suzanne, if I can take up 10 more seconds, that's a brilliant idea. And
grand challenge.
And it's an idea, but if there's somebody on this chat box who can organize that and put together a paper on that, I know that Keith and I would be happy to help back them up on that. But putting together a paper of what are the needs of the trainees in regards to research integrity, and when we see things like this. So maybe the trainees need to get together, the attendees need to get together, have a little virtual meeting and say, how do we address this? And then I will do my best to try to get this into an appropriate journal with you all.
Yeah, that sounds great. We've been collecting these for probably the last 4 workshops. So really want to just see what these great ideas are. And we'll get in touch with you on that. And I see Doris already says she would love to help with that. So I guess we're about 3 minutes over. Thank you again, Lee, as always. Fantastic. And now I'm going to go ahead and introduce Dr. Keith Baggerly. Keith, we can see your slides now. And get ready for a really entertaining, and if you think Lee's talk was shocking, this one is equally, if not more shocking in a different way, or kind of similar lines. But with that, I'll turn it over to you, Keith.
Okay, thank you. Good morning, everybody. So just as a quick bit of introduction, I'm Keith. I'm a statistician by training. And I worked for about 20 years as part of the bioinformatics group at MD Anderson. And while I was there, one of the things that really got driven home to me was the importance of reproducibility in high throughput biology, because that's what I was working with. High throughput biology, I'm going to define as, do we have an assay that measures thousands of things from a single patient sample from which we're expected to derive information? That's what I was trying to do every day for years. And it's often hard to do. And I'm going to tell you about that. And actually today, I'm going to be telling you a series of stories about some of the things that can happen. Now, there is a question for me of why is this particularly important in high throughput biology? What is there anything that's different about this in the context of hundreds or thousands of measurements? And the answer is yes, there actually is. And the main reason that there's a difference is that, quite frankly, our intuition about what makes sense when we're talking about tens or hundreds of things, it sucks. We have no clue as to how things interact in this high dimension. And that presents a problem for us, for folks who are trying to use this, because I want to take these tens of thousands of gene measurements. I want to concentrate the information into them, maybe focus on just 20, and using some pattern I see in those 20 genes, I want to make a prediction about whether this patient will respond to therapy or not. But there's an issue, which is that if I don't believe that I can correctly identify or understand that, yes, the biology is really doing this once I see the 20 genes, then I've got to have some other reason for trusting it, which means that I've got to be able to check every step of the process by which I assembled that signature so that I can be sure that I can at least check those things so that I can trust the edifice, if not necessarily have a base level check of the final answer. Now, the reason I believe that our intuition sucks is that I've seen it. And what I mean by this is that I've taken signatures of 20 or 50 genes to PIs, very smart folks saying, hey, you know what? We've looked at this pattern of 20 genes, and if they go this way, the patients do worse. And they've come up with wonderful stories for me, which is they'll say, oh, yes, I can see how that works because this interacts with this interacts with this. And they construct a story right in front of my very eyes. The problem is that on occasion, we have accidentally given incorrect lists of genes to PIs and still had them construct viable stories right in front of our very eyes. That's why I don't trust our intuition. So that's a problem. But fortunately, if we can understand or if we can see all the steps, we can avoid that. But this brings us to the next problem, which is if you go to the literature and you say, wow, this is a wonderful paper, I want to build on it, I want to do more experiments based on this. I know before I try to expand on this, I'll just make sure that I can reproduce what they did here. And as Lee alluded to, if you actually try doing this a few times, you may discover, and we did on more than one occasion, that the papers are not sufficiently detailed to let you do everything that was done. So that presents another problem, which is if you don't trust your intuition and you can't see all the steps involved, what can you do? Well, in some cases, if the paper is really important and you're not willing to give up on it, what you can do is what we did, which is we sort of developed a subfield of bioinformatics, which we called forensic bioinformatics. And here's how it works. You start with the raw data that they're working with, and this is posted at a repository somewhere. You then look at the results that they're reporting, and then you say, okay, what the hell must they have done to get from the raw data to their results, regardless of what the methods say they did? And that's often informative in surprising ways. And I'm going to tell you about this in the context of a specifically important clinical problem, which is, can we predict which patients will respond to which drugs? I know this is important because if a woman, at least when I was there, showed up at MD Anderson with advanced ovarian cancer, she would be offered standard frontline chemotherapy, which would be a platinum agent and a taxane, and we would offer this to her knowing that in general, it's going to help about 80% of the women we treat this way. They will have they will live longer. They will hopefully have better quality of life overall. That will help. But this implies, and this is true, we know going in that the other 20% of these women we're going to be poisoning for 6 weeks to no good effect. We do this because we don't know going in which 20% they are. If we knew which ones wouldn't respond positively, we'd offer them something else. And that's something we'd really like to do. And as against this backdrop, that a paper appeared several years ago now in Nature Medicine coming from a group at Duke, where it got a lot of attention because the authors claimed to have taken a huge step forward in addressing this specific problem. They've done it by making use of information available from brand new high throughput assays, which at the time were microarrays, things that would let them measure gene expression levels on tens of thousands of genes all at once. So great. So how did it work? Well, what they said is that what we can do is we can actually use microarray data and couple this with information or measurements we can make on cell lines to say something about patients. So what they did is they started with a particular panel of cell lines that has been maintained by the National Cancer Institute, the NCI, for the past 30 or 40 years, and this panel is known as the NCI 60. Now, why is this panel important? One reason why it's important is one of the ways the NCI figures out how different chemotherapeutic agents work is if you've got a new drug. Typically, the drug will get sent to the NCI and they will assay it for effectiveness against every one of these cell lines. And that data becomes part of the public record. So you can say, if I want to cut the growth rate of this cell line in half, how much of this drug is required? And what this means is that for each drug, you can then sort the cell lines from most sensitive to most resistant, and that can tell you something about how is the drug likely acting, but it can also give you some information about, can I tell the sensitive from the resistant ones apart in some useful way? Now, this is public information. Microarray data on these cell lines is also public information. So what they said is, okay, let's start with this hypothesis. We're going to pick a drug of interest. Let's say it's docetaxel. We're going to sort the cell lines according to their sensitivity to docetaxel. Then we're going to pick just the most extreme, let's say 7 cell lines at each end. And then we're going to look at the microarray data for those extremes, and we're going to contrast them gene by gene using immensely sophisticated statistics like 2-sample t-tests. And we're going to take, say, the top 50 genes that we find that way, and we're going to say, that comprises the expression patterns we see in those 50 genes, those comprise the signature of sensitivity to docetaxel that we can infer from these cell lines. Okay, great. Now we're going to go to a microarray pattern from patient samples, and we're going to look at these 50 genes, and we're going to say, for this patient, is the pattern more like the sensitive group or more like the resistant group? If it's more like the sensitive group, we'll say, give them docetaxel. We think it'll work. If it's more like the resistant group, we'll say, try something else. And the reason this got a lot of attention is that they said, we've been able to make use of publicly available data sets to test this for 7 commonly used chemotherapeutic agents, and they reported obtaining great results every single time. What this meant for us at MD Anderson was that a whole bunch of labs got really excited and a whole bunch of clinicians got really excited because they said, we want to treat our patients better, and this gives us a way to do this. We want to use their method. And of course, that led to lots of people coming to us in bioinformatics because they said, we want to use their method, but their method involves math, which is scary, so we need help. Can you help us? And we said, wonderful, we'd be delighted to do so because that's what we are. We're numbers geeks. We're sitting over here in bioinformatics. We're looking for ways to help patients. So we think this should be a beautiful opportunity. And in particular, there's something else here about this study that was really exciting to us, which is that all of the data that they're using is publicly available. So we should be able to check every single step and make sure that we understand what's going on. So we said, let's try this. And in particular, we said, let's start by doing or checking something that should be easy. Specifically, if we know one of the drugs that they're using, let's say 5-fluorouracil, let's see if we can identify the top genes and see how well our list of the top genes agrees with theirs. We thought this should be easy because to accompany this paper, there were several supplementary tables and whatnot. And some of these said, for this drug, here are the genes we identified. For this drug, here are the genes we identified, and so on. And within the text of the paper itself, there were often several explanations saying, this signature makes sense because this particular gene was identified, and we know this interacts with this drug in this way. So all of this looked pretty cool, looked like we should be able to do it. So we said, all right, let's get started. So we picked a drug, 5-fluorouracil. We looked at how many genes they identified. Okay, they identified 45, we can do that. We identified the sensitive and resistant cell lines, and we contrasted the microarray profiles. I did the 2-sample t-test myself. I am a PhD level statistician. I trust my ability to do this. And we looked at their list of 45 genes and our list of 45 genes, and we said, what's the degree of overlap? Do we really strongly agree, which we hope we do? And it turns out that the intersection, the overlap of our lists was 0. We had no genes in common whatsoever. And that was surprising. And we weren't expecting that. And we said, okay, what's going on? And it turns out that one of the things that goes on is related to the fact that once you start moving beyond 1 or 2 or 3 genes to thousands, at some point you often stop referring to things by name. You start using abstract identifiers or numeric things saying this is row 15, just to make sure that everybody's on the same page. And in particular, the microarrays that they were using were manufactured by the company Affymetrix, and the thing Affymetrix uses to label their individual genes are known as Affymetrix probe set IDs. So to illustrate why this is important, what I'm showing you now are the probe set IDs that they identified as being associated with the most important genes in their signature for 5-fluorouracil. On the right, here are the first few probe sets that we identify from doing 2 sample t-tests. And I want you to move your eyes from their list to ours, from their list to ours, from their list to ours. And well, here's one of the things that I would do, which is if I could see you all, I say, does anybody notice a pattern here? Because generally people do. They look at this and say, wait a minute, those lists are off by 1. And you're right, we have an off by 1 indexing error. So what's going on? Well, we went out and we managed to track down the software that they were using. I then wrote the documentation for that software because none existed before. And in the process of writing the documentation, one of the things that became clear was that the software required 2 inputs to work at all. The first input had to be a gene expression matrix, where you had genes as rows, samples as columns, and the samples had a header row saying, is this sample sensitive or resistant to treatment? Great, okay, that's the first input. The second input is a list of gene names in the same order as the gene expression matrix, but for their software to work correctly, this gene list must not have a header row. So if you're working in Excel and you cut and you paste and you leave on that header row, guess what happens? You get an off by 1 indexing error. And that's what happened here. Now, furthermore, what does that tell you about our intuition that this list got by the authors, the reviewers, and the editors at Nature Medicine, even though none of the genes in question are actually associated with the biology of interest? That's the case because our intuition about what makes sense sucks. We can mislead ourselves if we're given enough opportunities, and enough dimensions gives us enough opportunities. And this problem is present in the gene list for every one of the 7 drugs they looked at. So this is a problem. But while it's a problem, it turns out that this problem, if you think about it, really isn't fatal to what they're trying to do. What they're trying to do is they're trying to use the patterns of numbers, the gene expression values, to predict patient response. And the labels are things that got tacked on at the end to then hopefully give them an explanation for why it's working. But if the patterns of numbers still separate the patients into responsive and non-responsive groups, that's still something we can use. So can we do that? So we said, okay, before we get too excited, let's back up and let's take a look at the numbers and see if we can make predictions from those numbers, which are good enough for us to be happy using this. So that brought up a question of how good is good enough? And we thought about it and we said, okay, well, we're going to set as our first cutoff that we want to be able to get predictions that are pretty much about as good as they got for their paper. It was good enough to get them published. It's probably good enough to use. So that brings up the question of how good were their predictions? So I'm showing you some of their predictions in the top panel here. This is from the Nature Medicine paper. And what they did is for one of the data sets they had available, they said, we're looking at a cohort of 24 women who were treated for breast cancer with single agent docetaxel. And in this cohort, they predicted, or they came up with their scores, and they said, for most of the women who responded to treatment, were sensitive, that's the blue squares, most of them got low scores. That's good. And for most of the women who did not respond to treatment, were resistant, red triangles, most of them got high scores. That's good because there's clear separation between the responders and the non-responders. It's not perfect separation. So they say we only got 22 out of 24 right, but it's pretty darn good going in. It's better than we could do without it. So we said, okay, great, we'd love it. So we know that we can, here's a value to shoot for. But as we were staring at this, we had another deep statistical question that came up, and I'm actually going to give you guys in the audience a chance to think about this, which is specifically, how many sensitive patients did they have? How many blue squares are there in this plot? I'm going to let you guys stare at that for a moment. And it turns out that in this plot, there's 13 blue squares and 11 red triangles. That's what they can do. Now, why does this matter? Well, I told you before that all the data that they're using is publicly available. So actually, just to double check, we went to the source of this data set, which was a paper that appeared a few years before in The Lancet, and where the author said, if you want to tell our resistant from our sensitive patients, well, here's what we did with our 11 sensitive and 13 resistant patients. Not 13 sensitive and 11 resistant. And we looked at those numbers and we said, that looks odd and we're not too happy about that. So we said, what about a different drug? So we went on to adriamycin, and this one's, for those of you in the audience who are really ambitious, go ahead and count the number of red triangles, and you can send me email to me offline. I'll give you credit for it. But for most of those, just, are you willing to give me credit for the idea that there's more red triangles than blue squares? Because I think there are. And they said, okay, again, we're making predictions. Again, we're getting not perfect, but still pretty good accuracy. And this is still something we can use. Okay, so good. Problem. When we went and looked at the source paper for this data set, it was a paper that showed up in the New England Journal a few years before. And this paper in the New England Journal dealt with the treatment of childhood ALL, a form of leukemia. Why is this a problem? It's a problem because in this form of cancer is actually one that we know how to treat. We have a cure rate approaching 80%. Now, what this means is that if in this patient population, we have a drug which most of the patients are resistant to, we would never use it. So what actually happened is that the New England Journal folks thought that most of their patients were sensitive, not resistant. So we went back and looked at the data sets and the data that was being supplied. And it turned out, and I told you earlier, that one of the things that they needed as input to their software was a gene expression matrix with a header row saying sensitive or resistant. Well, what they were actually supplying was a gene expression matrix with a header row consisting of zeros for one group and ones for the other. But they never explicitly wrote down 0 should equal sensitive, 1 should equal resistant. And because they didn't write it down, in going from one drug to the next, in some cases, they got the order reversed. Now, what does this mean if you were to apply this clinically? It means that if their method works and you were to use this to prescribe drugs, you would recommend giving adriamycin only to the patients most resistant to it. And this is poor clinical practice, and this is not something that we recommend. So we said, all right, that's a bit of a problem. Now, this story going back and forth, this is not the result of 5 or 10 minutes of looking at the data. Actually, this is the result of the better part of 6 months of interactions by email, et cetera, between us and the investigators where we're going, we really like your idea. We like the premise. We'd love to use it here. Can you help us do it? We're the biggest cancer center in the country. If you can help us do it here, we'll affect more patients. By the way, are you aware that you have some of your labels wrong? And can you help us with this? By the way, are you aware these labels are wrong too? And then later on, it came to the point where we had to tell them, by the way, when we run your software on data using the labels which we think are correct, we can't get your method to work at all. What are we doing wrong? Here's our data, here's our code. Where are we screwing it up? At which point the authors at Duke said, well, this collaboration has obviously ceased to be productive, so we wish to bring this to a close. And we stared at that and we said, okay. And we collected our various objections and we sent it as a letter to Nature Medicine. And we waited to see what would happen. And a few months later, Nature Medicine published our letter. And alongside that, they, of course, published a rebuttal from the authors. And we were curious as to what the authors were going to say. Were they going to say, oh, we're so sorry, we'll fix it, we'll never do it, we'll never make those mistakes again. And that really wasn't the tone of the response. The response said that, unfortunately, the analyses by Doctors Baggerly and Coombs are deeply flawed. They did not use our methods properly. Our labels for adriamycin, that's the one with all the red triangles. No, our labels are correct. We've confirmed that. We've posted the raw data to our website to confirm this. Furthermore, since our initial paper appeared, we have continued to use the method in other high-profile publications, and we continue to get great results, so we must be right and they must be wrong. And we started this, and we were sort of going, well, okay, well, we're embarrassed because evidently we've just spent a bunch of our time shouting out in Nature Medicine that something's going on, and it wasn't going on. So we said, okay, where did we screw up? Well, they said their labels for adriamycin were correct, and they've posted raw data. So let's grab the raw data. We went to their website, we downloaded it. It was a big Excel table, about 20,000 rows, that's the number of genes, and 144 columns. The columns are samples, and the columns had 2 header rows. The first said, is this a cell line used for training the model, or is it a patient sample used for assessing how well it works? And is the sample sensitive or resistant to treatment? And when we looked at the samples that were nominally from patients and we counted the number who were resistant, that number matched the numbers of red triangles that they had. And we said, well, that matches, okay. Well, maybe they got it right. But if they got it right, where did we get it wrong? So we said, all right, can we find some way of separating the responders from the non-responders? So we decided to take a look at all the samples they gave us and just look at how well they associated with each other. And in particular, one of the things that we looked at was the correlation, the agreement between pairs of samples. And when we looked at correlations that were really high, we saw something that disturbed us. So in this plot, down here in the lower left corner, these are the cell lines, and all the figures we're showing here are correlations between one cell line and another. This line of red dots on the main diagonal, what is that telling us? That's telling us that cell lines are highly correlated with themselves. I believe that, that's good, that's what it should do. But then we come up here and we look at the patient samples. And this line of red dots on the main diagonal we expect, but these ones we don't. What's going on? Well, it turns out that their posted table of data has 122 columns of numbers saying that they're from patient samples, but those columns of numbers are not all distinct. Some columns of 20,000 numbers have been reused 2, 3, or even 4 times, and it's got to be from the same patient. Even better, right about here is one patient whose data was used for 4 readings, and that patient was labeled resistant 3 times out of 4. So we're pretty sure their answers aren't correct yet. So we sent this as a note to Nature Medicine. The Nature Medicine folks said, oh, well, the authors will make a correction. There's no need to publish your objection. And indeed, a year later, the authors published a reanalysis where they said, well, we've redone the analysis using only the 95 unique samples, and we still get great results. And this was surprising to us because when we counted, we'd only seen 84 unique samples, not 95. So we looked at the 95 they were using now, and of those 95, it turns out that there's only 80 that are unique, 15 are present twice. And of the 15 that are present twice, 6 are labeled both as sensitive one time and resistant another. So the correction of a correction still needs correcting, but Nature Medicine had said it was done, so we moved on. We moved on next to look at some of the papers that they, other high profile papers they had that they were using to expand on the approach. This one showed up in the Journal of Clinical Oncology, and it's trying to take the approach to deal with other drugs, specifically cisplatin and pemetrexed, which are widely used, amongst other cases, in the treatment of lung cancer. Again, they reported getting great results. And again, they said, this makes sense. Particularly, if we look at the signature we obtained for cisplatin, our signature contains the genes ERCC1, ERCC4, and some DNA repair genes. And there have just been a few papers showing up in the New England Journal explaining that this is how cisplatin acts on the body. So these make sense. So the folks in lung cancer came to us and said, can you check this? We'd love to use it if it works. And we looked at the data. And with some work, we were able to completely match their gene list for pemetrexed, which had 80 genes in it. That list of 80 genes we can perfectly match after we account for the off by 1 indexing error, which they are still making. For cisplatin, their gene list has 45 genes. When we account for the off by 1 indexing error, we can match 41 of the 45. For those of you in the audience who are good at subtraction, of course, that leaves 4. There are 4 genes in their gene list which are not identified by the numbers, either from our way of doing it or from their way, which includes the wrong genes. So what are the 4 that we can't explain? Well, the 4 probe sets that can't be explained this way are probe sets for ERCC1, ERCC4, another one for ERCC1, and another one for FANK-M, which is a DNA repair gene. And these are the genes that are used to claim biological plausibility. And we looked at this and said, well, that's annoying. But furthermore, there's actually something special, which is that if we look at these four, the last two have something that's special about them. Specifically, at this point in time, Affymetrix couldn't squeeze the genome onto a single array. They used a 2-array set. So those last two probe sets aren't physically present on the arrays that were actually used to do the measurements. They're on the other one. The other one was never used, as it means that 2 of their best genes haven't been measured yet. We thought this was a neat trick, and we sent a note to JCO saying, can you explain this? And JCO sent us a note saying, we're sorry, we can't publish this. We sent them a second note saying, could you tell us why? They sent us a second note saying, we can't publish this. So we moved on. We moved on next to the other paper that was cited as validation, and it's right there in their title, Validation of Gene Signatures. And this was a paper that showed up in the Lancet Oncology, high profile editorial, you name it. And what did they claim here? Well, they had extended the approach to deal not just with one drug, but rather combination chemotherapies. And this is important because we don't typically treat cancers with just one drug. We tend to hit it several different ways in the hopes that something will work. And in this cohort, what they did was they were comparing 2 treatments for triple negative breast cancer. And they were looking at cohorts and they were treating them either with fluorouracil, epirubicin, and cyclophosphamide, that's a 3-drug combo known as FAC, or with Taxotere, followed by a combination of epirubicin and Taxotere. That's TEP, there's actually just 2 drugs involved, but these are the 2-drug combos. And what they claimed is that if we had used their rule to allocate patients to treatment arm, as opposed to randomizing, which is what was done, we would have improved the response rate from 44% to 70%. That is a big deal. We are really interested here. And well, they posted the raw data online. So we said, okay, great, let's go grab the array data, let's look at it. So we downloaded the array data and we looked at it and we looked at it and we said, let's cluster the data. Maybe we can see 2 clusters, the ones who respond to this group and the ones who respond to this treatment. And when we clustered the data ourselves, we didn't see 2 groups, we saw 3. So why do we see 3? Well, actually, the raw data is sort of interesting, and these are Affymetrix files again. And one of the things that's interesting about them is that the Affymetrix files include, in addition to the gene measurements, they include metadata as header rows. And one bit of metadata they include is the day on which this assay was run. So we then plotted these arrays as a function of run date. So this first block is over here. These black circles, these are patients who received FEC treatment. The green X's were excluded from the final analysis for one reason or another. This second block, these are patients who also received treatment with FEC. And this third and last block, these are all of the patients who received treatment with TET. So what we have here is an example of perfect confounding of the experimental design. And you will hear more about experimental design later today. But what this means here is that we have a source of difference between the 2 treatment groups that's probably real, but not due to the biology of interest. And that's a problem. So we said, all right, well, we're not too happy with that. Maybe we can use something else. So what other things are there in this paper? Well, they've extended it to deal with multiple drugs now. How did they do that? Because from a mathematical point of view, we sort of understood how they were doing it with one drug. They came up with a number between 0 and 1, and they split it in half and said, okay, sensitive, resistant. How did they combine numbers for different drugs? So this is the math slide. And we went back to their Nature Medicine paper and looked at more closely at it. And that paper said, well, here's how we might do it. And they outlined the procedure that they might follow for a 4-drug combo. So we said, okay, now we've got 3 combinations. This one involves 4 drugs, this one involves 2, this one involves 3. So let's show you some formulas. Here's what they actually used to come up with predictions. So the rule that they were using for 4 drugs is the probability that a patient was sensitive to the 4-drug combo is given by the probability that they were sensitive to the first one, plus the probability they were sensitive to the second, plus the third, plus the fourth, minus the product of all 4. Now, if there are any statisticians in the audience, you're probably looking at that and going, wait, that's not right. And indeed, the authors realized that this wasn't right because all of the numbers, all of the probabilities they got using this rule were bigger than 1. So what they did, well, they said, oh, well, we know the biggest value should be 1 and the smallest one should be 0. So we'll set those and linearly interpolate everything else to fit. And this step didn't make it into the methods section. The reason we know what they did is we tracked down the Excel file to where they recorded the results, and we moused over the cell and said, oh, that's what they did. And we said, okay, we don't like this rule, but what did they use for the other combo? Well, if we think about a 2-drug combo, actually this rule would be okay for 2 drugs. It's the independence rule for independent events. They just generalized it incorrectly. So what is the rule that they did use for the 2-drug combo? This is the rule. It does give you a number between 0 and 1, that's fine, but the rule is different. And that was not something that made us happy. Now, what's the 3rd rule? This is the 3rd rule. I have a standing offer to buy dinner for the first person who can explain to me where this rule comes from, because we have no clue. But what I can tell you is that this is the rule that they used, which means that each rule is different. And to paraphrase a famous American, as a bioinformatician, I have a dream. And my dream is, if you are going to validate a bioinformatic approach in a new data set, is that you would keep the rule the same. They didn't do that. And that's something that disturbs us. But okay, we'll move on. Is there anything else that we can do here? Well, actually, if we're not happy with the design, and we're not happy with the combination results, can we at least say something 1 drug at a time? And we thought that we might be able to try that. And we looked at it and said, okay, can we do that? And the authors thought of this too and said, well, we think this is a neat idea. So if you want to tell our responders and non-responders to the FEC combo approach, actually turns out we can do a good job using just the results for Cytoxan alone. And they showed in their paper this ROC curve. And for those of you who don't work with it, trust me that this is a beautiful ROC curve. The closer you come to this corner, the closer you are to being able to say, with this cut point, everything below here is sensitive, everything above is resistant. Wonderful. Well, we took the data, we constructed our own ROC curve. This is what I came up with. It looks a bit different. And then we also looked at this and we said, there's something else that's perplexing us here. And this actually came up as a biological question, which came up when we started having discussions with some of the doctors we worked with, which is, there's a definition involved. And some of you may know this, which is, what's a prodrug? Prodrugs are used in the treatment of various types of cancer. And what prodrugs are is they're drugs that aren't active in and of itself. They're processed by your body into the active form. And as it happens, Cytoxan is a prodrug. It's processed by the patient's livers into the active form that works against the tumors. But this brings up a question. If you administer Cytoxan, a prodrug, to a bunch of cell lines which don't have livers, how do they exhibit differential sensitivity? So we went back and we looked at the cell lines and we said, we can't tell them apart. How did they identify cell lines to use here? And we looked more carefully and we were able to identify that the cell lines that they were using were indeed sensitive and resistant, but they were sensitive and resistant to pemetrexed. They got the drug wrong, but they got great results, so who cares? And we wrote up these various objections. We sent this off as a letter to Lancet Oncology, and Lancet Oncology says, well, you appear to be arguing about statistical issues to which, as everyone knows, there are no right or wrong answers, and as a result, we do not see the need to publish your objection. And we moved on. We moved on next because some folks who were working with us, who were working on glioblastoma brain tumors, said they're now working with temozolomide. Can you check this for us? And we started working with them, said, oh, sure, we'll try it. And we tracked down the data and we said, can we match the heat map that they came up with for the signature? And after some effort, we had to say, you know what, we can't do it. We can't get it to match. What's going on? Well, maybe one of their earlier papers can point out some step that we're missing. So we went through some of their earlier papers, and in particular, we wound up staring at this heat map that they published for cisplatin 2 years before. And we looked back and forth at this, and then we wound up writing a letter to the editors of both journals saying, one of these is incorrect. We don't know which one. We'll let you figure that out. And indeed, we got an email shortly thereafter from the editor of CCR saying, oh, the author sent in the wrong figure. They will supply a new figure. No need to publish your objection. And indeed, the author supplied a new figure and a new gene list, which didn't agree with each other and didn't agree with the text of the paper. It's a neat hat trick to have a paper nominally about temozolomide with a figure about cisplatin and a gene list for some other drug entirely. But that's what happens. And at this point, we've actually been doing this not for months. This is actually getting on to about 3 years, and we're sort of getting tired. So why do we care? Should we just stop? And it's at this point that some friends of ours got in touch with us and said, are you aware that the group at Duke is already using these rules to guide patient allocation in clinical trials? And we said, no, we weren't aware of that. So we looked more closely at the clinical trials and the reports in clinicaltrials.gov. And we said, well, in some of the clinical trials that they are running now, they're using signatures for drugs where, based on the data they've posted publicly, we think they have the sensitive and resistant labels reversed. We're not happy. So we wrote up our various objections from the past several years. And the first thing we did was send it to the editor of a prominent biological journal for informal comment. And he got back to us, and his response was, well, your story's too negative. Can you fix that? And we stared at that and we said, you know what, we really don't think we can. So the next thing we did is we sent it off to the Annals of Applied Statistics. That's okay, because everybody reads that journal too. And the response there was a bit different. I got email from the editor that evening saying, you know what, I sort of like this paper. I'm going to handle its review myself. And 2 weeks later, we got another email from the editor saying, you know what, we've simply decided to accept the paper as is. We've already posted it online. And that means that I'm sending more papers there. But one of the things about this is that now our objections are published. They're part of the literature, which means that we could cite it. And we could cite it in discussions with members of the press. And that meant that just a week or so after that our paper got published, the Cancer Letter ran a cover story saying, oh my gosh, in clinical trials being run at Duke University, biostatisticians at MD Anderson are saying there is the potential for patient harm. And unbeknownst to us, the NCI read our paper, looked at this story, and they looked at grant applications from the same group at Duke, and they said, we see the same mistakes here. So the NCI sent a letter to Duke saying, we are concerned. Will you reassure us that you're treating patients safely? And Duke said, okay, we're going to suspend enrollment in these clinical trials. We're going to convene an external panel of experts to look at this and decide if the science is sound. And we breathed a sigh of relief because while we were pissed that it made it to trials at all, having an informal panel, given that it's here, okay, we can live with this. And then we waited to see what would happen. And what happened next is that while this investigation was underway, while enrollment is suspended, the investigators at Duke posted brand new data to their website. What did they post? They posted the validation data that they used for cisplatin and chematrexid to prove that it worked. Specifically, they said they used 59 data samples from a larger public data set of 153. And they said, these are the ones we used. We said, great. We downloaded the entire data set and we said, let's check them. And in particular, if the samples are what they say they are in this cohort of 153, what you should see in this plot are 59 red squares, perfect matches, on top of this blue line. There are no red squares on top of this blue line. As it happens, there are only 43 red squares in this plot at all. For those specific samples, we can match the numbers they're using to a data file that's on the web, and they've got the sample ID wrong. They're talking about the wrong sample, which means that they've got the clinical annotation wrong. For these 16 samples, we have no red squares at all. Why not? That's because for these samples, they've already scrambled the gene labels so badly that these are no longer valid samples of anything. But what we can tell you is that every one of their validation samples is wrong 2 years after they started clinical trials. And we reported this to Duke and to the NCI and waited to see what would happen next. And Duke said that they were going to restart the clinical trials because they had finished their investigation and that had strengthened their confidence in this evolving approach to personalized cancer treatment. And we breathed a deep breath and we said, will you show us the data? And Duke said no. Duke said, while the reviewers approved of our sharing the report with the NCI, we consider it a confidential document, and a future paper will explain the methods. We're going to treat the patients this way now, but we will explain why it makes sense later. And we were not happy with that, but we did note that what Duke said actually gave us another option, which is that they shared the report with the NCI, which is a federal agency, which means that in the United States, it's subject to the Freedom of Information Act, which meant that a few months later, we were able to obtain a copy of the expert's report from the NCI pursuant to a FOIA request. And we looked at it and we said, this doesn't justify restarting trials. What the hell's going on? And Duke had no comment. And at this point, we were rather depressed because we didn't see that we could do anything else. And we were sitting around, and one evening I was sitting in my office. I got a call from a lawyer who said, you know what, we think there's some things you should be aware of. And I talked with him for a while, and I gave him the phone number, and I said, you know what, here's the phone number of a reporter you should be talking with, because I think he's going to find that really interesting. And about an hour later, I got a call from Paul Goldberg of the Cancer Letter, and Paul saying, I'm going to have fun. So why was Paul going to have fun? He decided he was going to have fun because it turns out that one of the investigators at Duke on these various clinical trials used to claim on his CV and on grant applications that he was a Rhodes Scholar, but the Rhodes Foundation disagreed. We know about this in part because Paul sent him a series of questions in writing saying, could you explain this? And Anil Poti, the person in question, said, well, maybe I should talk with him. After all, I was a nominee. And the fun thing is that Paul had CC'd a bunch of Duke deans when he sent the initial email, and Poti hit reply to all. So what had just happened is at Duke, you have had a member of your faculty admit in writing to a reporter that he's been committing fraud for the past several years and CCing a bunch of your senior administration as he does this. He was placed on administrative leave that day. This was a Friday. I got this story. I spammed a bunch of my friends and I said, I know a bunch of you have been talking about wanting to do something, write a letter. Now is politically the best chance you will ever have. The following Monday, a letter from a whole bunch of biostatisticians landed on the desk of Harold Varmus, the brand new head of the NCI, saying, would you please resuspend these clinical trials? Varmus called Duke and said, you're going to resuspend these, aren't you? Duke said, yes. Varmus called the Institute of Medicine and said, would you serve as an independent reviewing body? And the IOM said, yes, this is stinky. We'll take care of it. We'll start in December. But before they got there, before they got to December, something else happened. Specifically, in late October, Joe Nevins, the senior author on most of these papers, sent a letter to many of his co-authors saying, you know what, we may have to pull one of the papers. What paper might we have to pull? It's the one dealing with cisplatin and pemetrexed. Why might they have to pull it? It's because on closer examination of their database, it appears that 43 of their validation samples were mislabeled and they couldn't match the other 16, which was what we told them several months before they kept enrolling more patients. A few weeks later, Duke terminated the trials and announced that in retrospect, these trials should never have begun. Weeks after that, a cascade of retractions began. Poti resigned from Duke and things went on. What things went on? Lawsuits got filed. A misconduct investigation got launched. It launched a whole bunch of retractions. This actually was the very first story on Retraction Watch. It hit the front page of the New York Times. It hit 60 Minutes, and it hit a bunch of other places. And I'm going to play for you a clip from 60 Minutes to give you an idea of some of the things that were going on.
Dr. Nevins discovered that when the underlying data disproved Dr. Poti's theory, the data were changed.
It became clear that there was no explanation other than there was a manipulation of the data, a manipulation of somebody's credentials, and a manipulation of a lot of people's trust.
Manipulated data. These were not errors.
That's correct. It simply couldn't be random. It simply couldn't be inadvertent. It had to been based on a desire to make something work.
Is it a close call or is it abundantly clear that the data were fabricated?
Abundantly clear.
It is abundantly clear that the data were fabricated 5 years after clinical trials began. That is the wrong sequence of events, folks. Now, I want to tell you something. First off, I want to acknowledge that this data, this was a really screwed up situation. We don't see this many mistakes all the time. But I can tell you that we had seen each and every one of these component mistakes before, mix-ups and sample labels, because these are easy mistakes to make. We've gotten them in data sets at MD Anderson. Now at MD Anderson, we look at it, we say, oh, the data looks weird. Can you send us the raw data? We'll fix it. And if we have access to the source data, we can do that. But if what gets posted to the literature is an incorrect data set and you can't get access to the raw data, then you got a problem. And a problem can be pretty bad. And furthermore, right about the same time, when we were saying this is how bad it can get, a whole bunch of papers, including one from Begley and Ellis, were saying this is not an uncommon problem. which in particular led to the NIH and various others saying, we need to do something about this. So they talked about this in part because they said there's a lot of money involved. And let's look at part of that money loss. Why are they losing money? What's causing irreproducibility? Biological reagents and reference materials, this is, did you get the labels right? Study design, did you avoid confounding? Data analysis and reporting, did you tell us what you did? That's 90%, $28 billion. That's simple to fix if we know what we want to do. Furthermore, we were actually asked by Nature what we would want in papers when they were published. And you know what? We thought about it for a while and we said, we want to write up a description, we want the raw data, we need the code because written descriptions aren't good enough anymore. But really, there's a simpler way of describing what we want. Now is the point of a publication because that's when the journal editors have leverage. That's when they can say we want it, and that's when they can get quick action. Now, if we look at some of the things the NIH asks for today, have you identified your cell lines? Did you get the labels correct? Have you discussed the experimental design? Are you avoiding confounding? Have you thought about it? Have you discussed a data sharing plan? Are you thinking about how you're going to tell us what you did? And part of this is to point out that it is not necessarily impossible to do. They're really trying to get rid of the big errors, not the small ones. We, of course, tried doing it some of ourselves, saying, we know we can screw it up at MD Anderson. Are there things we could do better? And we've done things better. Furthermore, for those who want to learn more about this, we have done this as part of a short course for the GCC, for which the videos of me talking about this are online, and you can have several hours about me talking about how to define your workflow, how to use nice file names, and so on. So that's some of the things you can do today. But I want to tell you that there are reasons for hope. We're optimists because while a whole bunch of this was screwed up, I've been watching the data evolve for the past 20 years. And I will tell you the data quality and the tools for making it high quality are much better now than they were 10 or 15 years ago. So we can do it better. And I'm seeing us do it better. So I think we can get there, and it's moving the right way. Now, I wanted to toss out a question to those in the audience, which is what would you do? Because something that came out during the lawsuits, it didn't actually come up when all this was going on, is that there was actually a student in Anil Cody's lab who tried to raise concerns as all this was going down. And at the time, he was shut down at Duke. And that's the wrong sequence of events. It took several years for his concerns to be validated, but it happened. So that's just something to be aware of. Now, I do have to acknowledge that I didn't do all this myself. So here's a list of authors and things like that. And first among equals is Kevin Coombs. He was co-author of most of this with me. And I do have to acknowledge and thank one other group, which is you guys for having put up with me for the past 50 minutes. That's what I got for my main presentation. Thank you very much. Happy to take any questions.
Thank you, Keith. Great talk as always. We are a couple of minutes over, so I'm going to go ahead and dismiss, invite those of you who would like to go ahead and, except you, Keith. Those who would like to go grab a lunch to go do so, just you can turn your video and your microphone off. We'll be back promptly at noon to start the experimental design panel. Keith, there are some questions in the chat specifically about asking authors for their script. Are they obliged to do it? Is the some things about PAM 50, Mama Print. If you want to just kind of scroll through there and respond to those, that would be great.
Okay, I will try to respond to them in the chat and leave written evidence of what I have said.
That's right, it'll be there for good. Thanks again.
Okay.
See all of y'all at noon, sorry, at noon. Okay, welcome back. Can everybody hear me? Okay, good. I see Rovia nodding. So hope you had a good lunch break. We're going to get started with our experimental design panel discussion. I want to take just a couple minutes here to introduce our experimental design panel. And so I've got these listed in alphabetical order. So we'll just go in this order. If our panelists could unmute yourself and just introduce yourself, your institution, and a little bit about what you do in your research and kind of your specialization, if you will, in a particular type of experimental design. I know you all do several different types of experiments, but Dan, let's go ahead and start with you.
Sure. Hi, everybody. I'm Dan Harrington. I'm over at UT Health. I am your cell guy, your cell person, and your antibodies person. So we do a fair bit of 3D cell culture in our lab. We do a lot of imaging to follow. And so having good cells, solid cells, and good imaging and staining is important to us. So I'll be here to talk about those things.
Great. Natasha?
I'm Natasha Kirienko. I'm at Rice University, and we study host pathogen interaction using bacteria like Pseudomonas aeruginosa and Enterococcus faecium. We use C. elegans as a model and also do some with some research with human cell lines, but only your generic type of cell culture, nothing fancy like 3D.
Sorry about that. Okay, Robia?
Hi, my name is Robia Pautler. I'm at Baylor College of Medicine, and I work a lot with small animals of human disease, or mouse models of human disease, and also I do a lot of imaging.
Kurt?
Hi, I'm Kurt Zhang. I'm at IBT. So my area is most biostatistics and bioinformatics. So although I'm doing a little bit of animal studies, I have a wet lab as well. We're involved in statistical experiment design, clinical trial design, those things, and do a lot of bioinformatics analysis.
Great. Thank you. Thank you all. So I'm just going to tell you, I'm having kind of a hard time seeing the chat box. So panelists, are y'all able to see the chat box? Great.
Yes,
Yes.
Suzanne,
Yes.
there's actually a question from some of the panelists. They want to know when and how do they enter the breakout groups?
Yeah, we'll get to that when we do the breakouts later. We'll actually hit a button that'll put you in your breakouts.
Okay.
So you don't
So
have to
the-
worry
yeah.
about that.
So don't worry about it now, panelists or attendees, I should say.
Okay. So we're going to go ahead and get started. The first question that we're going to have is no, it's not advancing. Hang on just a second. Okay, here we go. Take a look at this question. When is it okay to exclude outliers? So I'm going to open a poll here in a second, but first just take a second to read through these. And then we're going to see what the group has to say, what the favorite answer is here, and then we'll turn it over to our panelists.
There's so many good ones.
Okay, so the poll should be open. Go ahead and start recording your answers. Okay, we'll take about 5 more seconds.
You're a very active group. Already 85% of people voted in 1 minute.
Not too bad, huh? Okay, so I'm going to end the poll and let's look at those results. So yeah, so the vast majority of you liked E with a second for B. And so I'm going to get out of this and turn it over to our panelists. So we liked E first and then B. Maybe you can talk about why some of these are not right. We did have a couple of takers for D.
And the gold standard, of course, if there is an error in lab notebook. And there are cases, for example, you're staining cells for a certain phenotype and suddenly add 3 times more dye. Or another good control that we're running, we always in a drug treatment, you running when you're testing new drug combination, but your single drug control suddenly doesn't kill. So likely a molecule just died during freeze thaws. So in a case when controls do not work and that's recorded, then it's okay to exclude the experiment. But important thing, what I'm trying to teach my lab members, even before you do the experiment, verify at what conditions are passing and what are conditions for exclusion. For example, if you study paralysis, say that control cohort should be at early stages. And if, let's say, every single organism die in day one, that's likely not because of condition, but some other contamination happened. So very carefully consider your variables, consider your outcomes, and write which parameters are acceptable based on your controls. And if your controls do not perform, then something globally went wrong. That's a good way to do it. There are also statistical tests when your controls work, but experimental variables some reason give weird results. There are statistical tests to see whether performance of a replicate is, let's say, 2 or 3 standard deviations away from the average. However, depending on how rigorous you are, people are different degree okay with it, but maybe Kurt as statistician knows more about that.
So as I'll turn it over to the other panelists and ask them to chime in. But again, if you have anything you want to add to the conversation, please enter that into the text box, or you can unmute your mic and just ask it.
So the question didn't say is this a human study, animal study, or bacteria study? For human study, I mean, this is very clear. So if you have to exclude some recruiter subjects, you need to have the exclusion criteria and the inclusion criteria. So you have to follow your protocol, and you cannot do by any statistical judgment and unless that's clearly stated in your protocol. So let's carry out this to animal study. So animal study normally most people don't have this really restricted protocol to say what animal to exclude, but I will highly recommend it. So yeah, you put some exclusion criteria, like when we say if you, for example, you do a cardiovascular disease study and you record the heart rate is about like 300. So okay, we can exclude. So if you put that in protocol, that will be wonderful. If not, then yeah, still, I mean, based on common science, just like Natasha said, so if the animal disease, certainly you can remove it. So the least we can recommend is, I mean, use the statistical criteria to say, okay, if the outlier is beyond the 3 dimensions, 3 times of standard deviation, then that's normally suggested, right? But that you have to be really careful to do that. So because this really depends on sample size. So suppose you have only small sample size, like 5, 6 animals, possibly I would never recommend you to use. But if you say you have 100 animals, just one of them outside of 3 times the standard deviation, so we're very comfortable to remove that. So this really depends on how good your experiment design you performed.
Thanks, Kurt. I think too, really key what Natasha said, making the decision before you do your experiment and analyze your data. You know, if I have this small number, we're not going to do it no matter what. We're only going to do, you know, having these decisions up front is really helpful. So I don't see any questions in the chat. Unless one of the panelists flags me, I'm going to move on to the next one. Okay, so how does the population of samples affect the statistic analysis? For instance, if I have 2 groups, each has 50 samples, the t-test shows they do not have the significant difference to each other. However, when I increase that to 200, the significant difference may be presented. Who wants to tackle that one? Just noticing that Lizette Rios is saying that GraphPad Prism has an identify outliers option and lists ROUT and Grubbs method. she wants to know, is this okay to use? What do you think, panelists? We'll backtrack just for a second.
I've seen this being used, but I think then it's important when you're writing a paper, write in materials and methods that outliers were excluded based on following statistics. And actually, right now, a number of journals wants to see all raw data, and there you can mark the samples that were excluded. I mean, my postdoc recently put heroic effort of having 300 lines where each line was independent experiment she did for the paper, and then another detailed table with every sample. So now I'm sure anybody who wants to recapitulate our statistics can do it easily. So as long as people know what you did and how you did it and have your raw data, I think it's valid.
So the raw data, including the outlier.
Yes. And I think I just want to echo her sentiments that you have to be very explicit about what the criteria were or are for exclusion from those data sets.
Okay, so let's move on to this one. Dan, you're smiling. You want to tackle this one?
Well, I don't know if I do, but I do. I want to add one more bit on just to say that I think Natasha hinted at this, that the moment where you added 3 times as much dye to the sample or something and it turns out awful, there's a moment where we're all tempted to just put a big X on the page and post on Twitter, you know, my life is awful, and you know, like, just move on, where it is really valuable to write up what happened and write up the conclusion and the bad result, and say, because you in 2 months may say, wow, if only we had that control of where we add too much.
You never know.
To
Right.
make it, to show the control of how it dies or something. Or that may accidentally happen, or you need to see how far you can push the assay. And so it seems awful, but it really has value to treat each experiment like an experiment and write up what was done and what occurred and what your interpretation is each time. Because you'll forget some of that 2 or 3 months later, and looking back on it gives you the timely perspective of that moment.
And actually, it's becoming even more rewarding. Now, people publish detailed protocols in a specific methods journal, and often there's a section, what can go wrong? And we sometimes on purpose did things like overexposing, adding too much dye, and then it will go, if this is your result, that's likely what you did. And these are possible source for common mistakes and how to fix it.
Okay, thank you. Who wants to say something about sample size here for this question?
So I think based on same, I mean, from first questions, so everything we should plan early. So if you want to do 50 samples you propose, and then let's do 50. And if you want to do 200, let's do 200. So that's the statistical principle, right? All the experiment design, all the data analysis method you have to put in front before the experiment is done. Okay, so the problem with this, you increase, when you test 50 is not significant, then you increase 200. So one of the very direct effect is you did the test twice, right? So this become a single test become two tests. So there's a multiple test problem. Definitely you need to adjust that. But it's beyond that, because there's a further question, like the 200, does that include the original 50, or is the complete independent set of 200? So then your adjustment will be different. So actually, even we think about the statistical way, I'm just trying to think about the way to adjust it. This become more and more complicated question. So yeah, as a principle, we have to put this in front, and we have to plan early. And that's all the question about your experiment design, how many samples you're going to collect.
Yeah, I just want to dovetail off of that. It's just like Kurt. So I think then by planning early, I think it's just like you're hinting at maybe people should do power analysis just to estimate, what sample size you are required to see the expected result. And I think some things that people approach me with sometimes, and maybe you want to comment on this since you are the statistician. What if you have no idea? You don't know what to expect. You don't know how to, what perceived difference or what possible difference you might expect from your results. How would you advise the trainees on this?
So, well, we all read a lot of proposal and propose something we never did before. So normally we have some idea. I mean, it's not come from nowhere. So maybe from literature we can find some people collect any preliminary data relative to a study, and well, we can say, okay, well, we fit different diet. We expect the increased weight. Body weight is 20%. So we can put all those assumptions based on that to adjust your sample size. Again, I mean, even you have a complete low idea, I mean, you still doesn't hurt you do a small pilot study with 3 animals and yeah, just test it. So yeah, that and certainly all those procedures is very accepted. I mean, when I read the proposal, I mean, never have the problem. So I say, okay, I assume the body weight will increase 20%, 30%, and the variability based on many of our other studies, and possibly we expect 10% of standard deviation. When we gave those based on that to calculate the power, I mean, I never have any problem.
Right. I definitely agree with that. And I think so it's just like to highlight some of your points too. It's just like maybe if you are proposing something new and you don't have specific data on those types of projects, but you might have past data on something similar or something that you observe in the literature, it is okay to say, okay, well, we would assume or we're expecting to see this type of difference. And based upon that, from our power analysis, we expect to use this number of samples for our study.
Yeah, exactly.
Okay, again, audience, if you have anything you want to add, please feel free to put that in the chat. And oh, we've got one from Doris. Is it valid to divide a known set of data into a hypothetical set to generate data and a validation set?
Doris, could you elaborate a little bit more upon what you mean about that?
Doris, do you want to unmute? And
sure. So if you have a set of clinical data that are which are in a novel field, is it possible to take some of the data to look at exploratory hypotheses about that data set and then use another subset of those data to test those hypotheses? I'm not sure if that makes sense.
It sort of does.
In a clinical trial with a given endpoint, if you're looking at something unrelated but might be mechanistic, can we develop a hypothesis based on what we see in a small subset of those data, unless it's validated in the rest of the data?
I think you can potentially do that, but I think you really have to guard against cherry picking to make sure you're not biasing yourself when you're defining which groups or what you're looking at. Does that make sense?
So I don't know, Doris, is this more relative to computational modeling? Because this is very standard approach in modeling. Okay, so when we do bioinformatics and yeah, like machine learning or AI, we have an AI model, and certainly we retrospectively have a study, and for example, we have 500 samples. We won't test our approach, test our model, and but this 500 is only data we have. Then how do you validate your model? So that's very common. We divide the data into 2 parts. One is the validation side, one is the training side. Possibly we put more data in training side than like maybe 300 in training, 200 for validation, and we can do cross validation. So we can do this many times. Each time we do the different division, okay, and use this to validate your model. So if the question is, yeah, for model validation, I think that's a very standard approach to do. But yeah, I'm not sure that Doris, that answer your question.
Yeah, Doris, let me jump in and maybe I can give another example too. So there's like brain imaging initiative data. So there are databases like Kurt was hinting at of let's say imaging data and from clinical populations. And anybody can access that data and you can analyze it however you want to. And so you can subdivide that to test a hypothesis if it's available. So yeah, it is possible to reuse data like that. Does that answer your question?
Yeah, I'm thinking more in novel fields where nobody really knows how to analyze the data and we're trying to come up with new ideas. But this makes perfect sense. And I think AI is going to start to provide, or it sounds like it's beginning to provide methods where that's done routinely. And so it'll be more acceptable than a manuscript or something.
Yep, I agree.
Okay, let's move on to the next one. Okay. So when doing an experiment, is it necessary to reproduce the results with different biological samples or technical replicates sufficient? Should it be 3 of each? What about qPCR? What about plating cells from one flask in different wells and treating that as a biological replicate? What do you think, panel?
Should we start by asking audience what do they think?
Absolutely. I think we can go ahead, but I would love for the audience to add to the chat or unmute your microphone. We can wait a few seconds and see if anybody unmutes.
Hi, my idea for a biological replicate is that it should be a completely separate instance. I mean, this is mainly working with cells. So a completely separate flask, maybe even done at a different time. And if you get a sample from that, that could be a biological replicate from another sample that you may have obtained from another day from another flask. That's how I think about it.
So you would say no to that last question, right? Can I plate
Correct.
cells? Okay. All right.
That's my thought.
Panel, what do y'all think?
I think it's a great start because biological experiments need to be biologically independent to make sure you don't make a mistake on cell dilution, drug dilution that will affect your results. So I even ask people to make drug dilutions independently, don't use stocks, pre-made mix between biological replicates. And of course, if you'll get your cell culture from ATCC, you'll get one tube, but you can right away multiply it, freeze multiple flasks, and then propagate these lineages for a while until they become independent. So if there is some sporadic mutation happens in one of them, others are not affected. And those who work with bacteria know that bacteria adapt to things quickly. You really want to start with independent colonies coming from the plate and not do any subculture, because the goal is to capture scientifically, biologically meaningful differences and not some kind of sudden fluctuation that happened. And with regard to number of replicates, it's nice when covariance, so standard deviation divided by mean between biological replicates is under 10%. Some experiments are inherently noisy. If somebody did metabolic analysis with seahorse or some other techniques, there is lots of noise. And there are people who will do maybe 4 or 5 replicates. But of course, nobody will expect you to do 15 replicates and then suddenly find statistical significance. At some point, it will become p-value farming and not a reasonable reproducible science.
Right. I think an analogous situation with just looking at animal models is if you do histological assessments in, like, let's say in the brain, and you look at 10 different regions, you can't treat those as 10 different biological samples. It's just like it's 10 regions within one mouse. So you would have to do multiple mice, and you could collect multiple regions within that mouse, but you definitely have to have multiple mice. And sometimes you see people kind of masking that a little bit. So I think when you're writing up your data into your manuscripts, these types of details need to be very explicitly clear.
So with regard to the question of, is it necessary to reproduce results with different biological samples, the panel would all agree, yes. Are technical replicates sufficient? And we'll lump that with, are 3 biological and 3 technical enough, and should they always be performed? What does the panel think about that?
At least 3 when it's possible. In some cases, if you're getting cells from a patient, there may not be enough for 3 replicates. But then you explicitly say that I have sample limitation, but then 15 different patients with 1-2 replicates per patient. But being that stated explicitly. I also noticed qPCR, what determines different biological sample, and RNA from an independent pool of your organism, cells, bacteria, mice. So you can't take same RNA, do cDNA synthesis 3 times and call it a biological replicate.
So there's actually a question in the chat, and maybe Dan, do you want to answer this one? It's from Deepog. I would assume that the sample, i.e. cell lines, could be used again, but under different conditions or collection times, and not my area.
Yeah, I mean, that would be the route would be to have independent freeze downs or kind of flasks going that that biological difference can be developing over time as you put more media in one or in another, or it's a different part of the incubator, or as they different seeding densities, these things kind of prop up, and although we're assuming that they are the same kind of base lineage, there's going to be slight differences that reflect the real experiment. And I would respond to the 3 and 3 by saying, the 3 is a start, but if you know that this comes back to the power analysis, if you know that your signal is not going to be revealed with 3 replicates, then that's not enough. You'll need something more that speaks to the signal you're trying to obtain.
And if you're using something as simple as cell culture, you can set 6 wells per technical replicates. And that's reasonable and acceptable in the field. So 3 biological, 3 technical replicates would only overall be 18 wells. So you can still test multiple conditions.
It is always helpful to do a little bit more when you're at that kind of situation. If you're just doing an assay that can spend a little bit more, you can plate a few more. Things happen all the time where you bonk the thing into the side of the plate, or something happens that give yourself a little cushion too, that to account for the randomness of experimentation. Things happen.
Yeah,
go ahead.
I just want to emphasize once more, since both Letitia and Daniel talk about independence. So that's a key word to decide if it's a biological replicate or it's technical replicate. So biological replicate must be independent. So that's very important. So I want to stress it. And regarding 3 biological, 3 technical, I mean, as I already said, that really depends on the power, your power analysis. So even for technical replicates, many times we possibly think 2 or 3, even 2 replicates, like a qPCR, many times we possibly just perform 2. That's enough, but this still based on your standard deviation of the technical replicates. So I see a lot of cases. So when your standard deviation between your technical replicates is really variable, so you possibly need a more technical replicates, and all the technical replicates do is one is quality control to say your technical works fine, so you have consistently generate data and have small errors. And another thing is they give you the chance to more accurate estimate the value you want to estimate. So like qPCR, so you will not take one of those technical replicates as your data, you take the average, right? So this average give you more accurate estimate of your target value. So that's why, so sometimes you need a more large number, larger than 3 technical replicates. That's just give you a chance to more accurately estimate the value you want.
So Doris notes that this is ideal in an ideal world, but how do we get grants, et cetera, to pay for these? Or what do we do if the biological replicates just don't exist?
I'll take that and say, at least for the first half, you have a better chance, I think, of getting the grant to pay for it if you build in this kind of statistical assessment that is sufficient. I think, at least in my experience, it is more common to see somebody say, after the grant's funded, oh, that was just for grantsmanship. We just were writing that into the grant. You only do 2, or just do it once and do a quick repeat. And frankly, if you're writing it carefully and doing the replicates well as designed and following it through, then you do have the better chance to get the statistical significance and the true data that you were hoping for when you first proposed the experiment. I think the follow through is the hardest part sometimes.
I'd like to jump in after that too. So Doris, I think you also have to be careful about what type of grant you apply for. So R01s do require preliminary data. R21s are not supposed to require preliminary data, but you see a lot of reviewers still looking for it. And it should be noted too that a lot of the ICs at NIH, some of them are actually not supporting R21s anymore. So there are smaller grants, excuse me, called R03s. So if you don't have enough data necessarily, it's probably best to go for the exploratory R03 or R21 grants, so you can eventually build up to the R01.
Okay, let's move on to the next one. We've got another poll here. So how important is the rigor and reproducibility section in an NIH grant proposal? So it'll be interesting to see. Some of you may have an experience with this. Let's see. Okay, so I'm launching this poll. Go ahead and indicate what you think on the importance of this in an NIH proposal. We've got some for all of them. This will be a fun discussion. Okay, we'll take 5 more seconds. Okay, so let's see what the results look like. Wow, almost a 3-way tie between scored section, required but not as heavily weighted as other sections of the proposal, and an absolute make or break. So Robia, why don't you start us on this?
I would love to. Okay, so I do review for the NIH about 3 to 6 times for study sections, and I'm actually now a chair of one of the study sections, the SBIB 10 study section. It's an imaging study section, and it is a scored section, and it is definitely something that can make or break your grant. So it could really divide whether or not you get funded or whether or not you do get funded. Those of you who thought it's required but not as heavily weighted as other sections of the proposal, it's actually extremely heavily weighted. So this is something that as reviewers, we are required to address specifically in our reviews, whether or not this was adequate. If it's inadequate, you're going to get dinged. So Suzanne, is it possible to go to the next slide? I think if it's the one I think it is.
Well, the next one is just kind of following on that. How complex then should an experimental design be to be considered a good standard for NIH rigor of prior research? And I think that's a good question to lead us into the next slide, right?
Yes.
So
So
this is kind of gonna address all of that. You want to walk us through this, Robia?
yeah, I would love to. So this is actually a slide that I was given by one of the past SROs, and I think it really nicely illustrates what you need to include in any proposal. So not just R01s, but fellowships, R21s, R03s, anything that you send to NIH or even foundations, because a lot of them are focusing on many of these criteria. So the first thing that we'll go over is the rigor of prior research. So this basically is, why are you doing what you're doing? Do you have a good reason? And these types of things need to be posted or included in the significance and also in the approach. So basically is the prior research, which means your preliminary data or your past research or that research that exists in the literature support what you're proposing to do. And another way of looking at this, if there are weaknesses in the rigor of the prior research, let's say the existing data within the publications related to what you're working on, are there weaknesses that also support what you're doing or what you propose to do? And again, it does affect your overall impact score. I don't think you can see, here I am using my mouse, acting like you guys can see my computer when I know you can't, but I am circling the yes. Yes, it does affect your overall impact score. And what I would highly recommend for those of you who are working on grant proposals, use these buzzwords in your significance and approach. You say the rigor of the prior research is, and explicitly state it. So when I write grant proposals, my significance in the approach, I actually have small subsections where I that specifically have rigor of prior research. That way you can just spell it out directly for the reviewers. Now, the rigor of the proposed research relates more to what we're talking about here. So are the strategies proposed and do they ensure a robust and unbiased approach? So what you need to include, this needs to be in the approach section, and yes, it does affect your overall impact score. Can you convince the reviewers, yourself and the rest of the world, that what you're proposing to do will be robust and unbiased and also reproducible? So I think you just have to very, very clearly state out much of what we've already discussed, but also there's some other, whoops.
Sorry, let's see if I can get back.
Oh, here we go. So some things that they specifically look for too are, for example, considerations of sex as a biological variable. That does need to be included in the approach, but also I note it as well as in my vertebrate animal section. So if you're working with vertebrate animals or human studies, you do really need to include those sections. And yes, that does affect your overall impact score, as does authenticating your key biological and or chemical resources. You have to have a specific plan, and that's also included in the additional review considerations. It's not technically supposed to affect your overall impact score. Typically that's addressed after the scores are made. But if a reviewer is going through this and they see you haven't adequately addressed this, it will bring into question just how rigorous or unbiased will your approach actually be. So I do think that on some level, it does affect the overall impact score, even though technically it's not supposed to. But are there any questions from the group if you want to discuss?
And we should also note that we were going to send a survey out, and it'll have a link to all of these slides where the video will be posted from today. And this is one of the resources that will also be posted. So you'll be able to take a look at this.
I have a question about, hi, I'm Vishesh Yanananda from MD Anderson. I have a question about gender inclusivity in animal studies, since the NIH is really insisting on that. If we are doing experiments with tumors, like PDX tumors derived from a male patient, does it make sense to only test it, put it inside male mice and test it in male mice, or do we have to also include male and female mice? And these experiments, because certain kinds of these experiments, they have to be done in very expensive mice, it becomes a huge cost burden for us. And sometimes NIH does not allow that kind of expenses just because the funding just isn't there.
Right. So I think you just have to be careful how you craft your budget. But getting back to your question, you have to address sex as a biological variable. That's not just a stipulation that you absolutely must do all of your experiments in males and female mice, let's say, in the case of animal studies. But if you have a really good reason not to do, for example, your studies in female mice, you just have to state it, and then that should be sufficient. But if you don't have a good reason, then you have to include male and female mice, and then you have to determine if sex is a biological variable. So, for example, let's say you were injecting tumor cells into the prostate of male mice to look at prostate cancer. This is kind of like a rough idea. I'm not really a prostate cancer person, but that would be a really good reason not to include female mice, because obviously they don't have a prostate. So as long as you state that very, very clearly in your research strategy, and again, I would bold it, I would underline it, you could say, female mice will be excluded from these studies for these specific reasons. Again, as long as you have a good reason to exclude them, you're fine.
Thank you.
Yeah, I would build on this and just say that the feeling that there's not enough money to do some of the things we've been talking about so far, I think is built on a feeling that there's only so much money, and that's often true. But if you say, this is how much money we need, but we also need this extra amount for doing STR identification and verification of our cell lines, or some other kind of validation that you build into your budget and say, because we know that this is important experimentally and that the NIH values this, we're building on this extra money that's earmarked just for this portion of validation or additional experimentation.
Yeah.
I'll also, I'm sorry, do you want to build off of that?
Yeah, I just wanted to quickly note, Dan, that the study sections don't make the funding decisions. So they're just going to be looking at the science. It's basically the program officers, it's the NIH, it's the different ICs that make the funding decisions. So I think I definitely agree with Dan. You write your best case, you write out your budget. If you get cut, if your budget gets cut, then you just have to work to revise it, but still include what you need to include to produce rigorous and reproducible manuscripts and pieces of work.
I think it's also important to let people know that they can, though, appeal a decision if a budget is cut based on something like that and you feel like you need it for rigor. You can go to council.
Sure. I think it's a moment of human nature here to think about, or if we're kind of focusing on these grant ideas for a second, to say, there are people who read to the back in the budget and they'll make comments. Your reviewers will sometimes say it, and you're protecting your integrity of your research by spelling out very clearly why you would need to do this. And it's hard for a snotty reviewer to say they've asked for too much money here or there. It's not their purview to be commenting on it, and it's easier to defend when it's described in that way. I'll also talk about human nature by saying one of our answers earlier was, is rigor and reproducibility, is it a tiebreaker? Is it just kind of something that puts you over the top, all other things being set equal? And I think that leans back to the way we do science. We learn from the generation before us, and some prior generations may say, this stuff is extra, like that's really not that important, and you might have learned it in that way. And we're saying to you here, it is critical, it is part of it, and it isn't necessarily a tiebreaker anymore. And so the language that you put in that clearly identifies that you've definitely paid attention to these aspects makes it easy for your reviewer to find them. When you have a section that says sex is a biological variable, when you have a section that says rigor, I do the same thing that Robia just mentioned. I put the rigor very clearly so that your reviewer can find it very clearly, and they know that you care about it, and they can score you well when they've seen that you've paid attention to it. It's a generational shift for all of us. It's been around for a while, but not that long as these things go.
Thanks, Dan and Robia and Doris and audience. If you have any more comments, please feel free to enter those in the chat. Again, this will be available on the link that we'll send you on the survey. But let's go ahead and move to the next one. What are some ways to approach rigor and reproducibility with respect to computational modeling? So invite the audience to unmute, enter things in the chat. We'll give you a few seconds to do that, and then I'll ask Kurt to jump in and discuss if we don't hear anything pretty quick. Kurt, why don't you get us started on this?
All right. So yeah, regarding computational modeling, I already answered part of it early. So it's very general practice. I mean, we use validation, cross-validation, and the independence validation. So as the golden standard of validates computation modeling is to use independent validation. That means if you are able to, so you collect, for example, 500 subjects to do to train the model and test the model, and then you'll be able to collect another independent data set, like another 200 subjects, and use that to test the results, or validate the results come from this early 500 subjects. Then that's a golden standard. But many times we don't have that, so so we have to use the way like Doris described earlier. So like divide your data and do the validation within the data. So normally what we do is we call it cross validation. So like you have 500 patients recruited. So we divided them into 5, for example, and each time we select one of them as a validation set and another 4 paths as a training set. So use a training set, we use a model to get a funding, and use a validation set to test it, and we do this 5 times. So and at the end we take the average and to see how good your model is. So that's a common standard way to do. So but regarding the rigor and reproducibility, so certainly there's many other issues to consider. So it's not just validate, and for example, how comprehensive, how robust your model is. So if you are doing, you'll be able to use simulation data. So one thing you need to keep in mind is you make sure your simulated data is really comprehensive. So it's not just fit into some particular pattern and let your model just reach optimize into a local minimum, what do we call it. So we have to make sure your generated simulated data is really comprehensive and covered all possible situations. And yeah, so possibly, yeah, so certainly there's other things to consider, but yeah, in terms of rigor and reproducibility, I think those are possibly the most important things to keep in mind.
Thank you, Kurt. And Alex noted if you're using code to post your code, oh, sorry about that, in GitHub or some other repository like that. We can go ahead and move to this next one about data and code. How long should it be kept after the project is published? You want to jump in and take that one, Kurt?
Oh, yeah. I think there's a guideline, I mean, from different agencies. So federal agency, I think generally recommendations are 3 years, 5 years, possibly 5 years. But as a research point of view, we normally keep the data as long as possible. I mean, even lifetime. So all the research data we think is so precious, and especially, I mean, when you publish data, I mean, the audience reader, I mean, will trace back even after 20 years, 30 years, they possibly still have questions to ask back. So we should allow that to do. So yeah, but I think the trick part is when the PI leaves the institute, close life, or what are you going to do? So certainly if the PI, I mean, stays in academic, I think that's PI's responsibility. So still keep the data and in the institute removed out too. But if the PI left the institute, I think the data is not the PI's has ownership of the institute also has ownership and should make sure the integrity of the data. So I think the best approach is possibly the PI should discuss with the institute, make a plan how to store the data and keep it as long as possible.
Okay.
So comment that I was going to make, grants are given to the institution. So it's not first author, it's PI's responsibility to have all data, but they're leaving, they need to give the data to the institution to be stored for appropriate lengths of time. And look, funding agencies specify, some will say 5-year limit. In some cases, they may ask for 7-year limit. So you need to be aware when you're submitting a grant, what are you required to do with the data. And NIH, at least NIH does audits. If you are audited, you're required to show all of the data and audit. I've seen what it can do to the lab. So the best you can do is to keep the data and hope that you can survive audit. Lots of Texas are funded by CPRIT. CPRIT also does audits. My grant was once audited by CPRIT and they found everything in place. So you don't need to do anything crazy to go through audit. Just do the good standards of bookkeeping, lab notebooks, electronic data, financial documents, keep everything in place. It's good for you, even if nothing bad happens. Well, not but even if nothing unexpected happens, but that it certainly makes your life during auditing much easier.
I'll add to that, Natasha, that we're going to hear from Melissa here in a few minutes, that if you're doing any kind of drug or drug development research where you're going to need FDA, there are really, even though it may not be hard, most people don't do it. Melissa's going to talk to us a lot about this particular subject of data and how to archive it and make sure that you're prepared for those things. So we'll have that talk coming up here in a few minutes. Okay, I don't see any other questions on this particular one, so we will move on. Okay, another poll. For assays that are very sensitive to user technique, how do you ensure that the results are reproducible across different labs? So I'll go ahead and open up this next one and invite you guys to tell us what you think. The poll is open. We're not getting as quick of a response on this one as we have. Here they come. Takes a minute to read through them. Okay, we'll leave it open for about 5 more seconds. Okay, so again, kind of split almost evenly with a slight preference for if results cannot be reproduced by different labs, they should not be published, or repeat at least 3 times in at least 2 different labs. But we should have a good discussion here because we had more than one for all of the answers. So which one of you wants to start on this one?
I can tell you that from one of our experiments, which involves intracarotid artery injection for inducing brain tumors in mice, it is an extremely difficult technique, and it really requires a lot of training, and some people are extremely good at it. Some people just cannot get it. And again, it's user skill that really determines how good an experiment, how an experiment turns out in the end. So in that instance, I would say skill is extremely important, as it is important for many physicians who perform fantastic surgeries, and a lot of patients would want to go to that particular physician who does it best. So I'm thinking in terms of reproducibility for animal experiments in the lab. that kind of situation, if that kind of a situation turns out, and you just have to mention that, that this is a skill associated technique, and a skill has to be built to an extent to be able to reproduce such data.
Panel?
Yeah, I think some of the animal studies can involve some very sophisticated methodologies. Like some of the stereotactic injections, when we're just doing like, let's say 10 nanoliter injections of contrast agents, that can get require some skill. But just thinking on a more broad type of level with animal studies, you just have to be so careful working with the different animals, because like, for example, if you're working with C57 Black 6, I mean, there's subtypes of the C57 Black 6 mouse strains. There's the N's and then there's the J's. And we had some issues because we worked with one over the other, and we found out that one actually harbors a mutation that affects oxidative stress, which totally threw us for a loop. We tried to figure it out for a year, and we weren't sure why we weren't able to reproduce some studies. And it turned out it was just simply that we were using the wrong background strain. But other things that can also impact reproducibility across labs when you're working with animal models is just like basically where the animals are housed. They're very, very sensitive to the housing. They're sensitive to the diet. If the chow is just even slightly different, that can certainly impact your study. So I think including as much detail as possible is critically important. And sorry, I was trying to read the chat, and then also because Charlotte just said something to me. But you just, you do have to be careful. I think reproducibility within labs, I mean, that's also sometimes of question. Like we've worked with collaborators who are chemists who might have one graduate student who makes the most phenomenal MRI contrast agents, and we're very, very happy. They graduate and they move on, and then the next generation of graduate students can't make something that's comparable. So I think just making sure that personnel are adequately trained before they move on is pretty important, or maybe even going to the point where when individuals reach out to you and ask for training, or if they can observe or they can visit your lab, I do think that it's important to accommodate that.
One comment I have, where the technique has to be reproduced by different lab before published. We've been in a couple of cases when we couldn't reproduce published data. When, for example, they will say there is only difference at the induced level, and we see clear difference at the basal level between two strains, even without any treatment. So the difference attributable to basal level and not to the treatment. We repeated it lots of times, and in a supplementary put all the raw images, and we published it because I was confident in what we were saying. So at the lab who published controversial data, at that point, PI changed institutions and wasn't interested in repeating their prior experiments. So we couldn't have it proved or disproved by their regional lab. So I think it's important to be confident in what you're saying and have all the data available for people who'd be interested in reanalyzing, but then go ahead, proceed with publication.
So that kind of addresses Kara's question, that is, who is the burden of proof on in these situations? Does this imply that if other labs can't reproduce results, the fault is on the lab rather than the user error of other labs? And I know that a while back, some people were proposing this second site validation, which means you've got to have funding for, twice the experiments. I just want to ask the panel real quickly before we end this section, because we've got about a minute left. Is anybody actually doing second site validation? And if so, how do you do that?
Well, I would say we've built it into an SBIR grant, for example, that we're doing. So we are the academic site. Our industry partner is doing similar experiments, and we did not build it in a detailed way. So I mean, in a rigorous way, but we did it in a it was kind of a sub aim to be sure that there was parity between the sites, but not in the full detail way that you described.
Okay, well, so panel, I'm going to ask y'all to go ahead and keep looking at the chat. It's been a good discussion, but we do need to move on with our next talk, which is Melissa. Thank you, panelists, and thank you, audience, for a great discussion. And I encourage the audience, if you have more questions, to continue to add those to the chat. I do want to introduce Melissa, who's going to give us our next talk. She is a perfect speaker for an afternoon talk because she will make you feel like it's first thing in the morning because she's so energetic, and it's so fun to listen to this talk. As many times as I've heard it, I learn something new every time. And without further ado, Melissa, if you want to go ahead and share your slides, we'll go ahead and turn this over to you.
All right, let me just make sure that I get it in the right view.
I can see it. Just needs to be full screen.
Right. Okay. Can you see it now?
Perfect.
Great. All right. I have my little timer going. So I'm going to try to get through this in about 30 minutes. So thank you so much for the invitation. Again, it's just such a stellar group of folks. My charge here is really to try to make a discipline agnostic presentation. So this is a little bit more tricky than it seems. But you have all of the scientific experts that you just got to sort of data mine this morning. So what I am going to talk to you about is data management and resource sharing. Just a 30-second background on myself. I'm a director of regulatory operations down at the University of Texas Medical Branch in Galveston. And my primary job is a translator from translating science to regulatory language, which is a big job. So most of the scientific community that I work with are the infectious disease community that are working with high consequence pathogens. So those are the diseases that we need biocontainment level 3 and level 4 to study and to work with. And that data that comes out of there is a lot of times an animal model supporting product development, particularly in the last 18 months to 2 years around SARS-CoV-2. And so a lot of that data then ends up being submitted to the FDA for a regulatory review and a decision to go into humans. And that data translation, if you will, is a completely different language. And so the base or the root of that is just to really make sure that we're documenting thoroughly and completely and in a way that folks understand. So I'm hoping that I can translate some of those lessons here today for you. So I'm going to talk about the basic principles and guidelines. I've just spent a couple of minutes there, and then the bulk of my conversation and my talk today is on the data lifecycle. We'll go into a data quality and integrity, which I hope to give you some tools to walk away with. For those of you who have kids at home, I will have an exercise that takes about 30 seconds, and they are welcome to participate. It'll make it a little bit more fun. And then we'll do a quick breakout for a case study. So there are 4 stakeholders in robust science, at least from my perspective and for this talk. It's an interesting link to an article. So all my slides have the links embedded in them rather than at the end in case you'd like to read anything further. This is an interesting article about institutions that are falling asleep at the job. You can see the researchers looking at them like, help. And I don't want to bash the institutions today. I don't want to put them on the spot. It's not fair. They're not here. But what I really want to talk about is, let's start out by looking at the funders and the journals, right? Because they have a responsibility to robust science as well. So in 2014, NIH got together with Nature and Science, and they had a workshop on reproducibility, rigor, and research of findings. And this was really primarily the journals that were the most common places that you would find NIH-funded investigators where they would publish their results. And the common opportunity here was they really wanted to look at what can we do as a scientific and publishing community to enhance rigor and reproducibility? How can we help and set some standards around that? And the outcome really are a set of principles that they were hoping that a lot of journals and other funding agencies would agree to endorse and adapt. Looking at the outcomes, the principles and guidelines, then there's a rigorous statistical analysis, which you've talked about or you've heard about this morning. Transparency and reporting, so making sure that you're disclosing everything. You heard some inclusion and exclusion discussions earlier. I'm going to touch on that too. Data and material sharing, considering established best practices for things like images, with Robia talked about biological materials, animals, getting journals and professional associations and societies to endorse these, and even journals to adapt these guidelines. So my goal or my charge today is to really drill down a little bit more into this data and material sharing. And for data and material sharing, this really means that these data sets need to be available when it's ethically appropriate, right? Sometimes we can't make them available in their entirety if we are dealing with patients, like the HIPAA rules, right? So we understand that sometimes they have to be stripped of those identifiers, and that's okay. Those are conditions for sharing. But we can be asked to share our data during a manuscript review and also upon publication. So this happened to me in 2019. I was actually publishing an article, or submitted to publish an article on health security, and it was about an educational program that we had stood up with the FDA. And the reviewers actually asked me for some of the raw data behind some of the summary tables. And so, of course, I provided that. But even on something as I don't want to say simple, but it's not as complex, certainly scientifically, in terms of publication. So just even on educational design, folks can ask for that. And then recommending these data sets be in public repositories where available, encourage the presentation of all data, knowing that real estate in journals is very expensive. That means if they're machine readable, or if there's large data sets, it means supplementary or pointing to where that information is stored. And then requiring that these materials be shared after publication, and encouraging the sharing of software, particularly if you need that code in order for it to be legible or readable. And then just disclosing where you can find those things. So if you want to have fun in a lab meeting sometime, this is a really cool YouTube video. It's been around for a while from NYU. And I love this name of the panda. Dr. Judy Benign is an oncologist, and she reads an article that's published in Science, and she says, this is quite interesting. I think I could maybe use some of this data and help my patients. She goes to the lead author, and the lead author is this little bear here, and says, well, everything you need is in the article, which is not entirely true. So she tries to get copies of the data, and it goes down this rabbit hole of, okay, well, I should give them the data, but where is it? And then there's the code, and then there's the graduate student that's no longer in the lab, and the knowledge went and left the lab with the graduate student. So they're very real scenarios that I see play out every day. And it's kind of comical, but in a sense, it's real life. So if you really want to have some fun in a lab meeting, run this through and then have a discussion about it. Pursuant to those adaptive guidelines that have been adopted by several journals, these are it here, and there's a link. I noticed some folks have talked about these already, but they also have a data transparency, and that means you should have this disclosure about, are your data are they available? So it's not just in your grant applications, but it's also contingent upon publications many times. And are you willing to share your data, and what would those conditions be? So that essentially your results could be replicated. And in a lot of journals, they want to know if this could be replicated pre-publication now. Here's a nice example of CRISPR technology, which is something that has reams and reams and reams of data, right? Like millions of data points. And it's a nice disclosure at the end that says if all the underlying data for the sources, the figures are provided in supplementary, and then of course the code availability, the code is available upon request. So there's an example of a nice disclosure. So philosophically, it's good to really understand that resource sharing is a requirement of NIH. They are funded by all of us through our tax dollars. And so they feel like that each scientific group that they're funding really owes back to the whole of science. That's really our charge, is to advance science as a community and not just individually. And so because of that, there are those requirements. So we spend a lot of time talking about NIH, but they're of course not the only funding mechanism for many of us. So always go back and read. I find this a lot when I work with investigators here, even at UTMB. They get a contract from somebody, especially if it's private industry, and they let the legal teams or the Office of Sponsored Programs handle the contract. And they're just working verbally off of what the scope of work should be with another scientist at the group. And they completely don't read the contract. and they find that they haven't really delivered everything that's in that contract. So understand what you're committing to when you go after the funding. Resource sharing from an NIH perspective doesn't just mean data. It can mean samples, reagents, novel reagents like antibodies. It can be model organisms like a transgenic mouse model. But from my perspective, we're really going to talk about the data. The University of Pittsburgh library system has a really nice all-inclusive definition of data. And while I don't want to read all of that, the second half of this really talks about being intangible and tangible data. So the intangible data are things like statistics, the analysis that you're doing, and being able to reproduce and reconstruct that. You know, what was your prospective statistical plan? What formulas did you use? What were the outliers? What were the conditions for the outliers? And did you save all of that, or did you delete the data? And deleting your data is not a good thing. You always want to keep everything and just explain why you didn't use something. So that's everything from gels and blots and cell lines and reagents and a whole big gamut depending on what your scientific realm is. So where do we begin? I'm a visual learner, so I like to look at things in terms of visual cues. So I put together this life cycle. You can do a Google search. I'm not innovative here. This is very similar to what you'll see out in the published literature. But you really need to think about from the collection all the way through to the reuse. So that means when we collect the data or capture the data, when we store it and how we organize it, about the analysis of the data, where we're going to retain it, both while we're on a study, but also for long-term retention. What are our conditions for sharing and reuse of that? And then we've heard some talk about retention and disposition. It really is the principal investigator's responsibility to know that. That is a moving target, but also consider where you're working. So even within the state of Texas, there are state regulations for minimum lengths of time that we have to keep these records. So it's a lot to keep in mind. I know most researchers like to keep it forever, but when we talk about that, think about where that's going to be and what's the longevity and the protection of that. So I want to talk first about just a data management plan, and this is prospectively thinking. You know, research data management is a term that describes the whole data life cycle, right? So managing your data throughout this life cycle. And it makes the following assumptions. This is also from the University of Pittsburgh, from their library system. The data are a scholarly product. And if you think about it, even from a manufacturing perspective, that's really what you're producing are these data. Your experiments are designed around the collection of data. Then you're going to analyze the data and then evaluate that against your hypothesis to draw your conclusions. Did you prove or disprove? The product that you're generating are the data. They're fragile, they're easily lost. There's growing research data requirements, as you've heard throughout the day. And so therefore, good management helps prevent losses and errors and increases the quality of your analysis. So well-managed and accessible data allows others to validate and replicate your findings, and research data management facilitates this. So one of the things I say to do is just do an exercise, right? So a lot of your grant submissions require that you submit a data management plan. This is part or separate from your data sharing, could be together depending on what your requirements are. But prospectively think about what kinds of data you have. Are they descriptive? Are they computational? What are the formats for keeping these, especially if they're electronic records? What would be your mechanisms for sharing and your access? Would there be any provisions? Would there be any conditions for sharing? Would you ask somebody to make sure that they redact certain things? Or are there any IP requirements if you're working with business and industry? And then provisions for that reuse and distribution. And that prospective planning really allows you then to think about how you're going to collect this data. And I'd like for you to drill down and think about source data, which is my next slide I'm going to show you. What's the original capture of your data? What's the first point that it's either electronically captured or that you write it down as an observation? And where do you do that? What's the format? How much of it do you have? And one of the things that I think we forget about a lot of times is who's the owner? We turn to the PI, which technically is the correct answer. But if I have a question, and especially if I have a 5-year center grant with multiple sites, and there's multiple people and subject matter experts working on a particular project, who on each one would I go to if I had a question? And I think we forget about sometimes the most common things in front of us, and that's one of them. We'll sit and have a question and go, I don't even know who I should call. So that's something I would leave with you to think about. When we think about source data, it can come in all kinds of shapes and sizes, and I've worked with a bunch of different scientists in a bunch of different fields, and I get a different answer no matter what. If you're looking at the top left and you're doing a plaque assay, it could be the first time that you take a Sharpie and you write down how many plaques you see. Or if you're a microbiologist and you're counting colonies on a plate, on a blood agar plate, you might use a wax pencil. If you've got a point of care testing or a very simple, say, hematology instrument there in the center that's printing out hematology, hemoglobin, and platelets and the like, and it may not keep that electronic data, but that printout might actually be your source data because the equipment itself is not keeping that. Or you may have something that's more complex where you're actually capturing that image and it's on a computer somewhere, or it's on a program somewhere, or it's a real-time PCR assay that's actually doing measurements and capturing that somewhere. Or you're out in the field. I work with some scientists now that are out in the field in Panama, and they have an entirely different set of how they're collecting in the rainforest, how they're collecting their data, and they have to protect it, right? It's entirely different conditions than if I'm in a micro lab. So really think about where those first capture of information is. And if you're like me and you're in a lab, a lot of times the first time you need to jot something down, you're going to grab the first thing that's at hand. So some of you, I hope, are kind of smiling and chuckling and laughing because we've all done this, right? We grab a napkin, we grab a Post-it note. Well, first of all, you have a high risk that that's going to be tossed before you ever have a chance to put this in a lab notebook or protect it somewhere. Second of all, I've very rarely seen a napkin taped into a lab notebook like the gels are shown here in the bottom right. So that means somebody is transcribing it. And anytime we transcribe, those transcription errors start to become a risk in our research. Here's an example of a hemoglobin value and its simple transcription error. If somebody's data entering this and they take the 13.6, they have a sticky keyboard, they all of a sudden miss the 1, and now the normal hemoglobin has now been entered into the Excel as an abnormal 3.6. And that's going to impact my analysis, but also potentially impact my conclusion. So if we ever do a forensic analysis, as we heard this morning from Keith, we always want to go back to those source records because the simple error is to find, oh, it was a transcription error. It wasn't something that was deeper into the science. So when we think about storage and organization, think about where you're keeping records. Think about that transcription risk. Think about who has access to those records, particularly important for electronic records, right? So, who has access to those source documents? I worked with an investigator here at UTMB who does the CRISPR technology. So reams and reams and reams. I say reams, that's the old paper method, right? But lots of electronic data. And what he does as best practice is he takes an electronic file folder and he locks it down. And then he makes an exact copy of that. And the original document he only allows himself and one other person to have access to. And everybody else on his team works off the exact copy. So if something inadvertently happens or somebody's computer is wiped out or there's a horrible event, they know that original source data is there and protected, and they can always go back to that. So think about things like fire and hurricanes and temperatures and things like that. If you've got a written laboratory notebook, what do you do at the end of the day? Do you keep it out on the lab bench, or do you put it in a fireproof cabinet at the end of the day before you go home? One of the things I really caution about is the use of Microsoft 365, which is great for collaboration, but it has this really cool or not cool autosave feature. And if you have that on for Excel, and we saw Keith give an example of a copy and paste error, the minute that you enter something and then maybe that's 30 seconds later, you realize you put it in the wrong cell, that has already rewritten over your file. It's an autosave. So if you don't have some method of control over the changes of those documents, then you, just by trying to share and autosave things, have inadvertently erased some of your data. So when it comes to electronic data, think about audit trails, which allows me to know who's attributable, who, if I have a question, who I need to go to. Metadata, which allows all my data to be readable. It's all that backend information, which allows it to be readable. Security and encryption, software compatibility. True story, I was in a board meeting one time several years ago, and somebody downloaded Skype because we were trying to remote communicate with a board member that wasn't there. And the individual who downloaded Skype, it actually it disabled the copy and paste function in Excel on their computer. So that's been several years ago. I'm not saying that would happen now, but sometimes there are random compatibility issues that happen. Always have backups. Always think about, are your program updates automatic, such as Microsoft 365, or if you're starting to run overnight, like I did one time with the PCR run, started to run overnight, went home, came back, expected to be really efficient the next day, and realized that UTMB did an automatic software update, rebooted all the computers, and I lost all my data. So there are those real sort of life experiences that we need to think about. So think about even your data migration as it moves along. So I need to ramp up this a little bit. So here's an example of, I just pulled the left one from the internet. Think about standard methodologies for your electronic files. Try to come up with something in your lab that's consistent. Try to think about how would you consistently organize your documents from study to study to study. This is really good for graduate students who have to do their dissertations on a building of their research over time. But it's also good in thinking about if you have to apply for an NIH grant and I have to justify sort of my prior research, it's easier for me to organize, much less trying to organize for publication. For analysis, that's really been talked about earlier today. I can't emphasize enough about inclusion and exclusion criteria. There's so many times when things are very nebulous when I talk to folks, and they just kind of have this feeling about, well, that just looks wrong. It's just out, as opposed to thinking in advance, what are my criteria for excluding? So I've addressed that bias in my research. So here's an example of images and falsification of images. The Office of Research Integrity has a lot of examples for this. I'll just focus in today on that figure number 2. Generally, this is more fun if we're in an audience, but if you'll notice M1 and M4 in figure 2, those are actually the same image, only they're flipped. And so this actually happened in a publication. Same thing with the image on figure 1, the COMET assay. The control and the protein are the same image. They're just adjusted a little bit, right? And so this is seen a lot. So here's some answers and some examples of a Western blot that's been flipped, and actually a gel shift assay where someone took a lane and cut it out and was very good at whatever it was, right? At Adobe Photoshop or whatever, and copy and pasted it, in to make it look like there was an analogy there. And images have also been talked a lot about by Robia, but I have this cartoon here that's really nice, again from ORI, which says, please keep your original source file, right? We know that we're going to enhance these images. We know we're going to do that, but in this particular example, this contrast and saturation increased the resolution, but it eliminated the background cell. So that can be an issue, right? So always keep your unedited source file. Protect your data through the retention. I have also been part of an NIH Office of Research Integrity audit. I've accompanied some researchers who were unexpectedly audited by them. And I can tell you that the auditor said, show your data some love. Like you just haphazardly document and throw it in a file, and you're not caring for your data when that's really the basis of all your conclusions. And so I thought that was a really good comment. Think about those conditions for reuse. It can be simple as, I have a small data set, I'll email it to you. It could be, it's in this online repository. Don't forget about instructions. I think that's another thing that people forget about, is they'll point folks to an online repository, but they won't share the instructions. This happened to me again when someone was really good about pointing me there, but I didn't know what to do with it. They had 3 or 4 different files and I didn't know how they organized it. I wasn't sure exactly how to deconstruct that data, and some instructions would have been really helpful. So here's an example of a data sharing plan for those of you who really want to see one. Again, NIAID within the NIH has a lot of really good tools. If you are applying for an NIH grant, there's really a lot of tools there that you all can leverage. So now I want to switch over to data quality and integrity. I just have a few slides about this, and then I'll wrap up, and then we'll go to our case study. Data quality, I like to look at it from the perspective of how FDA likes to look at it. This was originated by Stan Wolin at the FDA many years ago. Data quality, really look at it through the lens of, at any point, are your source data attributable, legible, contemporaneous, original, and accurate? So attributable means who? Who do I go to? Who's the one who recorded it? Legible means I can read it. So either I can read it and it's not a scribble, but it also means, is it legible in terms of code? Contemporaneous. If I ask you today, what did you have for dinner last Wednesday night? Could anybody come up with that, right? Unless it's an anniversary or a birthday or something special, nobody remembers that granular detail. Same way goes in the lab, especially if you're balancing a lot of things, like you're applying for a grant and you also have a postdoctoral fellow and you also have your research. So as soon as you can write it down, the better your memory is, the better and more accurate and complete it's going to be. And then retain those originals, those source data, or at least an exact copy. Whereas data integrity is, are your data complete, consistent, enduring, and readily available? So now I want to do an exercise. So everybody grab a pen and just any kind of piece of paper or something that you can write on. I don't really care what it is. And I'm going to put an image on the computer, and I'm going to give you 30 seconds, and I want you to describe it in as much detail as you can. Remember, it needs to be attributable, legible, contemporaneous, original, and accurate. On your mark, get set, and go. 30 seconds. I know you're scribbling. I hope it's legible. Five seconds. Okay, stop. Now, what I generally do when I'm face-to-face is I just ask people to start calling out their descriptions, right? And if we had more time, I would have you do it in the chat. One of the most interesting discussion items is, what is this little guy eating, right? Is it a strawberry? Is it a carrot? And it gets to be kind of interesting just from a subjective standpoint, right? So there's variability in research just in our observations. What we're trying to do is control as much as we can for the research to be reproducible. So let me ask you, was this attributable? So did anybody date and initial it so that I would know it was contemporaneous if I was keeping this over time? If you didn't, then please go back now and initial and date it. You can do this in the, right? Nobody's going to collect this. Nobody's going to make fun, right? Okay, so when we talk about dates, this is a really common one, especially for working with international collaborators and or if we have international students that are in our labs here in the States. Individuals will write dates down differently. If you are using the month, like MAY, it's not a problem. But if you're using numbers, this could be a problem. Today's May 27th. If I write 5/27/21 or 27/5/21, probably don't have a problem. But if it was May 7th and I wrote 5/7, some people would interpret that as May 7th and others as July 5th. So this is a very common thing. That's a really easy thing to correct when you're trying to reconstruct studies. People generally ask me about lab notebooks. I inserted this slide just as a really quick pros and cons. This is my opinion and what I've seen. The pros of lab notebooks, electronic notebooks, is that it really allows you to get organized. It allows you to more easily collaborate. You can customize forms so that you don't miss data. This is another really great thing, right? So you don't inadvertently miss a data capture that was part of your original design. It's searchable, another great thing. Has an audit trail, so I automatically can capture who did something. And most of these have an exportable format so that you can actually share these with your statisticians, because of course we're not doing our own stats, right? Okay, laugh. The cons are the cost, the sustainability cost, the system administration behind the scenes. Who's going to keep this going? Who's going to make sure it's updated? What are the compatibility with other systems? What about any other, like a Mac versus a PC? What about software updates? And would that impact the data that's already in there? Do I need to data migrate over time? And what happens if it's discontinued? So I tell people, just make sure your data are exportable to some type of long-term archivable, readable format, such as PDF or HTML. So I'm going to close by saying this is a really cool reference, the WHO Handbook on Quality Practices in Basic Biomedical Research. The mantra is, if it wasn't documented, it wasn't done. Without documentation, the process is meaningless. Essentially, there's been no study. You can't count on your memory. And the older we get, and I'm a baby boomer, so I can say that, the harder it is to pull all of those facts in our very cluttered brain. So just to reiterate, data are all intangible and tangible data. If we go back to this Begley-Ellis article, the one that had the 89% irreproducibility rate, these are the four things that the ones that were confirmed had in common. They incorporated controls into their experimental design. They authenticated their reagents. They prospectively addressed investigator bias, and they described their complete data set. So think about that. So here's an example of communication. I'm going to miss the March 3rd Friday call-in. I think, oh, it's Friday, March 3rd. But look at this on March 19th, March of 2019. March 3rd was actually on a Sunday. And what this person actually meant was it was a reoccurring teleconference that happened on the third Friday of each month. So miscommunication happens everywhere. Closing thoughts: get organized, think about data stewardship throughout the life cycle, come up with a data management plan, prospectively plan, implement Alcoa. And now we're going to go to the case studies. So I'm going to stop sharing and I'm going to turn it over to Suzanne, who's going to put us in breakout sessions.
Right. So actually,
right?
Shannon's going to dismiss us into breakout rooms. You should have those cases sent to your email. So have those ready to go. And we probably won't take the whole 10 minutes. We'll probably take about 7 or 8 minutes. So jump in, identify your reporter and the recorder, and remember to write those down, everybody's thoughts. Shannon, you can go ahead and put people in their breakout rooms. Okay, it looks like we're all back.
All right, all back. Let me share my screen again, just so I can make sure we're all talking about the same thing. Can you see it?
Yes.
Great. Now what's going to happen is it's going to lock and not advance. Okay.
Are you locked?
Yeah. That's crazy.
Okay, well, if you want to quit sharing, I'll share mine and you can talk.
All right, let's do that.
I've got yours up.
Perfect. So if we can do that, then I think what I'm going to do, because we're at the sort of at the end, here's the case study. Excellent. And what I would like to do is I'm just going to call on a random number. And if you're part of that group, just tell me your thoughts. There's really no right or wrong answer. So I'm going to call in group 8. So would somebody from group 8 please tell me what you guys talked about and decided would be your conditions for sharing, if you could share.
Sure. So I'm happy to take over to be the spokesperson for group 8. So essentially, we talked quite a bit about kind of multiple layers of this, even from blinding within the study to make sure that there's as these things are going forward, there's no knowledge besides the PI and having unique identifiers. But from the reporting and sharing of this data point of view, we talked about making sure that any sort of rare or unusual characteristics are actually redacted. So if there's participants that are over a certain age, that information is not disclosed, to make sure that individuals within specific groups are not given that identifiable information is not there. We also talked about potentially for some of this information, not even sharing it at anything less than an aggregate level, just to make sure that there isn't the ability to further point out individuals within the data set, depending on how it's used, to make sure that we prioritize that identity of the participant over the research kind of utility as well, and make sure we're protecting their personal health information.
That's excellent. That's really great. I appreciate those comments. And what I've done here and what Suzanne is showing is I pulled this off of the NIH website simply because they have a lot of good case studies, but I really like that one of these included a commitment to destroying or returning the data after the analysis are completed. And I think that's fine, right? So there's no right or wrong answer here. The point is that you as the PI have to think prospectively about what do I need to do to be responsible about the data and what would be my conditions for sharing. So if NIH has this charge about sharing data, what would those conditions be? Including maybe even an agreement with the PI. One of the other things I wanted to point out, which I think so thank you, Blake, I really appreciate that, great job. One of the other things I also wanted to point out was if you have any type of collaborators with your research and you're doing conditions for sharing, that you might even want to think beyond this particular case study and think if there's any other acknowledgments that need to be included besides yourself, right? So if you're sharing the data and somebody's going to publish and they're going to say, I'd like to thank Dr. So-and-so for providing this data, you know, is there anybody else that really should be mentioned in that? And so I just say be respectful of your colleagues as well. So I'm going to end there, lucky number 8, right? I only called on one group, but I hope that you guys enjoyed the presentation. I hope that it provoked some thought. If you have any questions, email me afterwards. I'm available and happy to chat. And otherwise, thank you very much. And I'll turn it back to Suzanne for Stacy to wrap up.
Thanks, Melissa. Appreciate it. All right. So yes, we'll turn it over to Stacy now, who's going to talk to us about publication and reporting. Stacy is from University of Houston, and she's been doing this with us from the beginning. And it's been fun to watch this presentation evolve over the years. And so without further ado, I'll turn it over to Stacy.
All right. Thanks, Suzanne. Everybody can hear me, right? All right. So sorry, guys. I have 3 screens going on, so I'm going to try to keep an eye on the chat as we're doing this. So thank you very much for sticking with us today. And the last thing I want to talk about today is talking about some of the more behavioral aspects, right? Moving from avoidance behaviors into transparency into research, right? So I want you, uh-oh, am I frozen? There we go. Okay. So I just want you for just a brief minute, you can close your eyes, you can just kind of like relax for a second. And I want you to think about time points or examples in which you personally have exhibited avoidance behaviors such as procrastination with respect to publication and or reporting in any of your current or past projects. So I want you to think about it. You don't have to disclose. If you want to share, you can go ahead and put that in the chat, but it's not required. So when we're thinking about these events, right, think about the why behind that, right? It's really important to understand why you did that so that we can actually break that cycle and move forward, okay? So typically when we have these behaviors, this is typically an avoidance motivated behavior, right? And so our behavior is when we distract ourselves by doing a task that's from doing a task that's associated with an unpleasant emotion, typically fear. There's different types of behaviors we might exhibit, right? So it could be complete avoidance, where you just no show at a presentation, you don't submit your documents, or you just quit a responsibility, right? So that's complete avoidance. Another form could be escape, where you leave early from a commitment, you abruptly end a talk, or you hide in your office. I've definitely seen faculty members that have decided to hide in their offices before. And then there's partial avoidance, right? Where we daydream or dissociate, or we avoid eye contact. Okay, so there are different aspects or different behaviors that are avoidance behaviors. A lot of times the underpinnings to these are things such as worry, anxiety, panic. And these behaviors actually don't reduce our anxiety. Actually, what they typically do is they fuel it, or they cause difficult situations to snowball, right? Where these situations get out of control. right? And so we want to stop these behaviors. We want to acknowledge when we're doing these behaviors, and we want to be able to change it. Okay. So usually we're trying to do something to create momentary relief. The number one way we do this is procrastination. Here's some funny comments or some comics. Field Guide to Procrastinators. We can all identify who we are in there. I'm definitely a snacker. And sometimes I'm also the cleaner, and sometimes I'm the list maker. So these are different ways we can procrastinate. And here's another funny one. The 4 horsemen of procrastination, particularly in modern times. We nap, we snack, we go in social media, get lost on Twitter, Instagram, Facebook, whatever your social media platform of choice is, or we attempt to do minor chores in order to do things like get away from our writing or what we need to be doing. So just some funny cartoons to bring some levity to this, right? So when we're avoiding our tasks, we're doing something to bring momentary relief, right? Avoidance motivated coping is not necessarily data fabrication or falsification. These are extremely maladaptive behaviors in which you never collect your data, you make up your data, or you illegitimately alter your data. These are some of the extremes, and we should not be doing these things, right? This is not good. Examples of these extreme cases, we definitely talked about this. Dr. Ellis talked about this morning. You can see these cases on the Office of Research Integrity website that I have listed here for you. So why do we exhibit these avoidance behaviors in publication and reporting? A lot of times the underpinning emotion behind it is fear, typically fear of failure, as well as some type of pressure, right? So there is pressure to have a high impact publication, whether that be in the CNS journals or a different journal. There can also be lab or peer pressure within. One of the things that was talked about also this morning are these high impact labs or these high pressure labs. And so that can definitely contribute to this fear and this avoidance behavior. There can also be issues with visa and employment pressure, feelings like you have to perform in order to maintain your visa status or your employment status, right? So these are some of the underpinnings. Other aspects that can contribute to avoidance behaviors, such as difficulty with time management. One of the things that, especially when I was a junior investigator, particularly a student and postdoc, I didn't really understand how much administration there really is as you become more independent as a researcher. And there's a lot of, sometimes something just happens very suddenly, you have to take care of it, and that takes time out of your writing, right? And so there are these chores that you have to do that can just chip away at that productivity time. And sometimes our deadlines for publication and reporting can create conflicts with other commitments, such as family commitments, teaching commitments, service, travel, and so forth. And those are all under time management. Another aspect that can feed into these avoidance behaviors is the feeling of a lack of progress, right? So often we feel like we haven't been productive enough, which can be paralyzing in and of itself, right? And so there's always that comment that you should be writing, you should be writing, you should be writing. Okay. Sometimes there's also difficulty in acquiring what you need, whether it's supplies or just information that is needed in order to complete a project. And sometimes progress really can feel like a moving target, right? You feel like you're never actually meeting your goals. So when all this happens, there are impacts to your life, right? You can have missed deadlines, you can have delayed publications, you can have late reports to regulatory and funding entities, which is really bad. And all this can impact your promotion tenure or other career advancement opportunities, right? And so when you procrastinate, it makes easy things hard and hard things even harder, okay? So we want to avoid this, right? We don't want these negative outcomes. So when we're talking about publications and products of research, we're typically talking about peer-reviewed publications in top journals that may require open access agreements for data produced. That's definitely been a very big aspect of this afternoon, talking about that. But there's other products, right? So there's conference papers and podium presentations, abstract and conference posters, right? Those count as products of research. In some fields, technology development or techniques can be considered products of research, as well as patents, patent applications, whether they be provisional patent applications or non-provisional patent applications, or even licensing agreements, right? Especially if you are in some of the bench science aspects and engineering, patents and licensing agreements are definitely considered products of research. There are other forms of products of research, such as databases, physical collections, softwares, instrumentations, things like that, right? And so it's not always just a publication that is considered a product of research, but there's this larger body of work. So that's something to consider, right? So maybe you didn't get that paper in Cell or Nature or whatever, but you've had conference presentations, you've had maybe some patent applications, right? So those are all products that are moving you towards your goal. So who does the work? Typically your primary investigator and collaborating investigator are your senior authors, but it's really your postdocs, your research assistants, your grad students, possibly even undergraduate students and lab techs. They're really doing the brunt of the work in the lab, right? So when we're talking about science, we're talking about team science, which the PI supervises. And then the PI, this actually allows the PI time to spend on other projects and commitments, many cases administrative commitments. And most of the work in the lab is done by our junior scientists and trainees. And I point this out because avoidance behaviors exist at all levels of science, right? And so sometimes we have feelings as graduate students or postdocs, like I'm not making progress, I'm not making progress. And I had a conversation actually like this with one of my, or one of the graduate students in our program about 2 years ago. And it's like, no, this doesn't necessarily go away. We deal with this all the time, right? So, and it let her, basically open up communication and talking to her fellow students about how she's feeling and coming up with some strategies to deal with some of these avoidance behaviors. So when we're talking about publication and reporting in science, there's different types of reporting that you might be responsible for. Could be reporting to your department, center, or university for annual reports. It could be to your IRB, or Human Subjects Protection Committee. It could be to IACUC, or Animal Research Oversight. It could also be to a federal regulatory body, such as the FDA, or some type of funder, right? Whether it be a federal level, NIH, NSF, or so forth. State level, example would be CPRIT. Local foundations like the Dunn Foundation, or major private foundations. So examples could be American Heart or American Diabetes. And when we're trainees in particular, we don't understand the amount of time that this can really take from our science, right? And so I show this graphic right here. When you're a postdoc, the vast majority of, and when you're a student, the vast majority of your time really is into developing your skill sets, developing your knowledge base, and being in the laboratory. However, when you transition to becoming a primary investigator, that time, it just gets fractured. And a big part of that has to do with basically the breakdown of what our responsibilities are during our hire. So for example, at the University of Houston, our breakdown is typically about 40-40-20. So 40% research, 40% teaching, and 20% service. And then from that, it gets further fractionated, right? And so you need to be able to manage that complex call for your time, essentially. Talking about research performance progress reports, when you're reporting to many of these entities, a lot of entities have taken up the format that has been required by NIH. And these reports are done on an annual basis. Usually only the primary investigator or a noted delegate can initiate them. And there's a lot of time that goes into this and a lot of effort. Majority of the work is probably going to be done by the PI, and it is typically a formal submission. Hopefully your institution will have administrative resources to help you with this, but that's not always the case. Always ask if there's somebody that can help you with this in order to alleviate that administrative burden. It's possible that it might not happen, but at least if you can get somebody else to help you, it can definitely help free up some time. Required components of these reports tend to be accomplishments. You have to list the major goals and objectives of the project and whether or not these goals were achieved. If there were any training and professional development opportunities during the project, was there any results disseminated, and how do you plan to accomplish goals during like the next reporting period, especially if there were issues. COVID is a huge issue, and that's something that many of us have had to deal with. And coming up with strategies to achieve those goals in this very unusual period. Typically, you're going to be indicating if you had any publications or products, and that includes preliminary data. If you have any patient or animal recruitment and safety reports to file. Sometimes you have collaborative efforts such that your project may be distributed across multiple locations, and so it could take time to get all the required components in. So always build in time to do that. And you might be asked if you are making an impact with your research in the desired areas. Sometimes that's a requirement, sometimes it's not. Like I said earlier, in these reports, project changes, challenges, and problems, they will ask about that. If you had to change your approach for any reason, anticipated or actual problems and delays. And if any of these changes have significant impact on expenditures. Over the past 14 months, we've definitely seen that with COVID, and there's just been a lot of people trying to figure out how to manage this in their grants and how to move things forward. You also have to do financial and budgetary reports. Those will be required. And if there's any modifications and or future plans, you will have to present them. All right. So we're going to pivot a little bit now to talking about different aspects of publication and reporting, some things that you should be looking for when you are doing your science, and things that these reporting entities, whether they be federal, whether they be manuscripts, whether they be your own peers, things that they're going to be looking for that you're doing in order to help science grow. So you need to be reporting on, are you doing randomization and how are you doing that? You need to be reporting on blinding, single, double, triple blinding, if there was allocation concealment, if the conduction of the experiment was actually blinded, or if the blinding was actually due to the outcome. You should also be reporting your sample size and power analyses estimations. You should be, basically as transparent as you possibly can here. And the statistical details of computations should be reported. I have an example of one that I've actually produced for a recent grant application. So here I actually show power for every aim and measures that are of interest under these aims, differences between two groups, and what the minimum power is and what the minimum number per persons per group would be. But you could do this for anything, basically just to ensure that you're making sure that you are powered across all of your aims. That is one thing that you can actually come under fire with under grant reviews in particular, or even possibly preprints, if you are underpowered for some of the questions that you're asking. Okay. Other core reporting standards include data handling. When are you going to stop data collection? Do you have inclusion exclusion criteria for your data or for your sample? How you define outliers and how they're treated? What your endpoints are, how they're defined, how missing data are going to be treated, and replications, right? And if it was replicated under a range of conditions. So these are all things that are core reporting standards, whether they're in publication or even potentially for oversight documents from granting institutions, from federal institutions, from your own institution. So these are all things that you have to basically work on and know these things, right? And that can take time away from the laboratory. Okay, so we have these hurdles, right? We have these challenges, right? So there's all of these things that we have to do. And then we can finally get our project up and running. But then we have issues with buying instrumentation, getting our participants, our lab animals in, getting our supplies, time, funding, pandemics, right? There's all these different things. And so we can have lack of progress due to such difficulties. And that can be really, really hard to deal with, right? Sometimes you have difficulty organizing information by the stated deadlines due to time conflicts, communication challenges, or just unforeseen events. And then lastly, if you don't have a lot of institutional support, it makes a hard job even harder, right? So some PIs need to fill out these reports and this information completely and submit it entirely, right? And so a lot of time can be devoted to understanding what information is required, especially without administrative support. And if the wrong pieces are submitted, you then spend even more time going back, figuring out what was submitted incorrectly and then correcting them, right? And there could be some negative consequences. So hopefully you have institutional support to do this, but I definitely acknowledge that some of you may not. So if publication and reporting is so tough, why do we bother? It's because there are negative consequences for not doing the things that we're supposed to do, right? It could be withdrawal of funding. It could be an inability to support current and future projects as well as current and future trainees. Also an inability to achieve promotion, tenure, and career advancement, okay? So Doris, yeah, to answer your question, when you basically power to the weakest aim, simply. So if PNR is tough, why bother? You know, it's not just about consequences that we feel personally to ourselves, but also there are significant consequences to the evidence base, right, and to our fellow scientists, right? So if we are holding back in terms of publication and reporting, there are certain biases that can be introduced into the evidence base. Probably the most commonly known type of bias is reporting bias, right? That's the one that's most commonly introduced, right? So where we selectively reveal or suppress information or the outcome of a study, where we report some results, but not all of them, or we spin unexpected or undesirable results, right? Okay. So that's absolutely something that can happen. Other types of biases, publication bias, which is non-publication of results, right? So we had a failed study that didn't yield positive results, so we're not going to publish it, right? So a lot of time and effort was spent into doing something that isn't going to yield a product. Outcome reporting bias, this is also known as expectation bias, which was once again selective reporting of results or modifying your hypotheses to fit your findings, right? And you're less likely to report adverse outcomes such as negative findings, right? And so you can definitely have issues with this when you're talking about like drug trials, device trials, things like that. Other types of biases include citation bias, a tendency to cite positive findings more frequently, and that leads to the perception that certain interventions are more effective than it truly is. And it's definitely results in overrepresentation of positive findings. There's also location biases, which refers to the journal publication and its impact factors. So think about Cell, Nature, Science, whatnot, right? So studies with positive and significant results tend to be published in your journals with higher impact factors and published in journals with better access, right? So these are your index journals. Okay? So we need to be aware of that. There's also language biases, such that publication of positive findings tend to happen in English, where non-significant results may be published in non-English language journals, which definitely will impact your meta reviews and systematic reviews. Time lag bias is another form of bias, where there's rapid publication of exciting but not necessarily full and complete results. So there can be a delay in publishing negative or non-significant findings, right? Sometimes PIs will attempt to do this if they want to try to boost their productivity under a specific grant. You shouldn't do this, though. Please don't do this. And then lastly, another major type of bias is your funding bias, right? So this is the tendency of a scientific study to support the interests of the study's financial sponsor. This is essentially an expectation or outcome reporting bias. Some sponsors do require PIs to waive the right to publish findings that don't support the sponsor's interests, which once again leads into publication bias and outcome reporting bias. And some of these studies are actually flawed from the start based on the influence of the funder actually on what actually gets done in the laboratory. There are major companies and entities that are trying to avoid this by basically disassociating themselves with study design and trying to be more clear about their conflict of interest, which I think is a great thing. But we're definitely, this is definitely an area that there's a lot of gray area and definitely needs a lot of work. So how do we address bias in PNR? Other than trying to report in a timely manner, report in a complete manner, right? Try to build a case. So if you have negative results, try to build a case in which the negative results actually provide a positive impact. Don't suppress results, particularly negative results. Don't rush or delay publication, but also don't dual submit. That's a thing that we are much, much easier able to detect now than we were 20 years ago. And don't publish the same results in more than one manuscript unless there's a very good reason to do so, like a new type of analysis and whatnot. And when you submit now, they will ask you if this is a dual publication situation. Also, carefully consider the audience in the journal in which you are applying to make sure that the fit is good so that we're not dealing with huge time lags because it didn't go to the right source. Try to balance the story you tell with respect to citations. And rigor and reproducibility guidelines and the journals that adhere to them. There are a huge list of them on the NIH website, and there's a link that I've provided on this slide to that. What else could you do? Don't withhold critical information. Make sure you discuss authorship and team expectations early on the process so you don't have issues later on. Trying to figure out, okay, who's going to be the lead author, who's going to be senior author, and so forth. Just try to have those out, those conversations early. Make sure you're organized as much as possible. I know that's a challenge for some people, but also make sure to set realistic goals for yourself and your team. And just realize that you might have to reprioritize depending on unforeseen circumstances, right? Above all, communicate with your team. So when you detect that there is something going on, with a team member, it's generally better to check in with that person or have somebody check in with them than to try to wait it out, particularly if there's something going on in terms of like visa stress or health stress, or just go ahead and reach out to your team members to make sure that they're okay. Because if you don't, situations can snowball and get out of control. What else can you do? Make sure that you are reporting on things like randomization, blinding, sample size estimations, and data handling, and look for these things in the literature you use to build your studies, right? To echo the last talk with Melissa, please make sure that you're using Alcoa principles to manage data quality. Make sure that your documentation is attributable, legible, contemporaneous, original, and accurate. And make sure that you're maintaining proper data integrity with complete data sets, consistent documentation, the medium is enduring, and the information is readily available. All right. What else? You should know the strengths and weaknesses of your team. You should familiarize yourself with the expectations of your editors, your review boards, your funders, your institution, right? So know what they need of you and when they need it of you. Know your deadlines, keep track of them. Know what information you need to meet those deadlines for your reporting, and know your resources, right? So do you have that administrative support to help you with things? And if you don't, make sure that you're building in time to deal with that, right? Okay, so all that being said, I have a challenge for you, okay? A new year, a new you. So we're gonna go into breakout rooms, and I want you to think about what you and your lab can do this year to improve your relationship with publication, reporting, and bias. So just come up with 2 or 3 resolutions with strategies that you think would work for you and your group to share in your breakout group. All right, Suzanne, I think it's time.
Shannon, you can go ahead and open rooms.
All right, everybody, welcome back. Thanks for working on this in the breakout rooms. We have a few minutes. So is there anybody who really had a really great discussion in their group and wants to share some of the resolutions and strategies that they come up with in their breakout rooms? I know it's end of the day.
Well, I'll volunteer. I was the breakout group 7. Well, not very extensive, but like the few things that we came up with were one, to actually like spend some time to discuss negative results. And so maybe this could be in regular lab meetings. And one of the things that was stated by Harman, one of our members, was that like it actually helps having regular weekly meetings with PIs, because if all you had was negative results that week, you'll at least mention that, because it answers the questions of what did you do with your time? And at least in that way, it gets discussed, even though it's not very exciting. The other thing was from the perspective of computational modeling. Ying mentioned that maybe it'll be useful to identify people, even if they're not necessarily people who work on the same thing you do, maybe other modelers, to ask them to use whatever you've developed, and without really knowing why or like, you know, a lot of background, see if they get the same results. And so that could be something to reduce bias, or like, because we might have certain hopes for our hypotheses, but the other modeler doesn't really care. That's not what they work on. The third thing I felt was that there is some inherent difficulty in like everybody in the lab being on the same page, because I do computational modeling, but like I might report on something every week, but that doesn't mean the people listening will remember those perspectives or values every week. And so what this inherently does is that like it basically creates situations where like where there might be different results to be true. And so I'm not quite sure like how to handle that outside of just, you know, discussing repeatedly and making sure like everybody in the lab has the same understanding of what's going on in their data. And that's it for our contribution.
That's great. Absolutely great. Thank you for sharing that. Does anybody, we have a few more minutes. Does anybody want to add on some ideas that they had within their groups? No, it's the end of the day.
I can go, sure. What the heck? Yeah, so in our group, group 10, I think, we had lots of great vigorous discussion on rigor and reproducibility and all these things. A wonderful group that I was in, and we had a very diverse group doing a lot of different things. So we had kind of 3 different thoughts from pretty much 3 different areas. So for instance, on the side of software and computational stuff, you can do things like using things like Docker and virtual machines for like making sure that everybody can run the same software on all the different computers that everybody has, which helps with this stuff. You can deposit your data in lots of repositories that are available to dump data on, like Sequence Read Archive, SRA, Dryad, and stuff like that. And you can always put like, you know, your code on GitHub and GitLab and things like that. And you can put all the code for all the tables and everything that you generated. On the side of like clinicians and working with patients in a clinic and stuff, you know, sometimes people will use recorders to like notate down everything that they've said after they've seen a patient. And that could always be gone back and got if you ever needed information from that. It can be transcribed, stuff like that, which can be helpful. And then finally, wet lab bench side. Someone in the group mentioned that you can, a lot of times there's like a bunch of different ways that you can solve the same problem. So maybe the more ways that you try to do it will help like with the reproducibility of it. So people can have a lot of different options to try to validate and reproduce what you did.
Absolutely. Yeah, that's all great, especially bringing in like the different perspectives of computational science and bench science versus clinical science, right? So these are some of the communication challenges we have. So your group did an absolutely great job bridging some of those gaps. So excellent. I think we're actually getting towards the end of the time here. So Suzanne, would you like to take the reins?
Yes. So if you will, first of all, I want to again thank all of our speakers, and I want to thank all of you that stayed with us through the day. I enjoyed getting to chat with some of you, and hope that you'll feel free to email me if you have any additional thoughts or questions. You'll be receiving a survey in the next couple of days. We really hope that you'll take it. It will be give you an opportunity to give us any lingering questions you still have. We have our Rigor and Reproducibility community. There's actually a board where you can post thoughts on our website. In the survey that I send to you, I'll also share the links for where the recording will be and the slides and any resources from today. And as I mentioned earlier, one of those questions, and I really want everybody to think about it, because this is kind of our grand challenge, is to try to think about how do we change the culture to where it isn't a publisher perish? I mean, or do we need to? Trying to think about strategies for that. So you'll have an opportunity to share that with the group. And again, I just thank you. I believe Shannon or Dawn is going to put the link for the post-assessment survey in the chat. You can go ahead and click on that and take it. Don't be surprised. It's exactly the same as the pre-assessment. We just want to see how much you improved after today. So yeah, thanks again. And we look forward to hearing from you. I guess I should say, once you're finished with the post-assessment, that's it. And assuming that you did the pre and post and submitted your cases in advance, you'll receive your certificate via email. Again, don't hesitate to reach out to me. I'll add my email to the chat as well. Thanks, Stacy, and thanks, everyone.
Thank you. Thanks, everyone. Have a good evening.